Hi, everyone, and we're really excited to have you all here today and joining us, and for taking some of your time on Saturday to come and listen to SciSpire. We're really excited to have you all here. And so my name's Alexandre, and I'm the chair of the Atlantic Student Research Journal this year. And I guess the person that's going to be leading the speakers and kind of doing the introductions is going to be Lauren McGrath. So I'll just hand stuff over to Lauren for her to kind of lead the way.
So my name's Lauren and I'm the junior chair, and I'm really happy to welcome you all to SciSpire 2020. So as Alexandre said, I'm going to be running the speakers portion, but before I start, I'd like to go over some housekeeping. We're going to hear from our speakers first for around 30 minutes each, and after each speaker, there will be a period where you can ask questions. So if you want to ask a question, we would ask that you use your raise hand function, and then I'll call on you and you can turn on your mic and ask your question to the speaker. And after the speakers, we'll have a session with some panelists where you can ask them questions regarding their professional pathways, research, or their specific projects. We will be leading the discussion at the start, but if you have any questions, you can feel free to raise your hand as mentioned before. And if there's no questions right now, we can start with introducing our first speaker. So our first speaker is Jake Stillwell. Jake is a recent graduate of UNB, where he earned a Bachelor of Science in Biology Physics with a concentration in cell and molecular biology. His research, which he will be talking about today, was supervised by Dr. Janice Lawrence. He uses genetic approaches to detect and characterize benthic toxin-producing cyanobacteria in the wall stock. There you go, Jake.
Cool to get started. Okay, so do I just share my screen with my PowerPoint kind of thing?
Yeah.
Okay, 1 second, I'll get this pulled up. Okay, so I guess first of all, thank you for having me at this event. It's a really cool opportunity, and I'm glad to see that you are giving undergraduate students the opportunity to explore and make connections with different research experiences. So for those of you who don't know me, my name is Jake Stillwell. Like it was just said, I'm a recent graduate of UNB. I studied biology and physics with a concentration in cell and molecular biology. And today I wanted to talk about my experiences with undergraduate research. So they've been great learning experiences for me, and I've had a couple of them. I'm going to focus mainly on my work with Dr. Lawrence. But when I was trying to come up with a title that really captured everything that I had learned through my experiences, I came up with something long and stodgy like a cumulative perspective on undergraduate research opportunities, how they have shaped my interests, clarified my career goals, and developed my skills as a scientist. But that was kind of a mouthful. So instead, I would like to call this, just kidding, Jake's Declassified Research Survival Guide. So what I'd like to do is talk about my different experiences with my different research opportunities and how they informed each other and how they informed what kind of research I wanted to pursue. So before I get started, I just would like to recognize that all of the research of experiences and projects that I've contributed to take place on the unceded and unsurrendered traditional lands of the Wolastoqiyik and the Mi'kmaq. I think it's important to recognize their continued stewardship over the land, particularly where the Wolastoq figured so prominently in my research in my community and in light of recent events. But beyond that, a little bit about me. Like I said, I graduated from UNB last year with a degree in biology physics. I'm currently working a couple of jobs, one with Dr. Lawrence, whose research I'm going to talk about a little later today, and also with a local STEM nonprofit called Worlds Unbound. So we do a number of different outreach events that are related to science, technology, engineering, and math. I actually have to run a virtual club after this, so I'll be jetting out after my presentation. And currently, I am presenting at the ASRJ SciSpire event to share my undergraduate research with you. So let's get started with that. So my first experience with summer research was after my first year, and it came in the form of an NSERC USRA, or an Undergraduate Student Research Award. So I was really interested in a whole bunch of different types of science and really just wanted to get my feet in the door any way that I possibly could. So I applied to work with someone who works in physics and mathematics at UNB, Dr. Vicar Hussain. He does a lot of research in quantum gravity. So the way quantum gravity works is, in physics, anything that's quantum deals with things that are very, very small. And that doesn't necessarily jive with a theory of gravitation and gravity, which oftentimes has weird, funky things happening when things are moving really, really fast. So we did a number of different problems and simulations that summer to try and reconcile these two pieces together. What I found was that there was a lot of computational work, and that computational work wasn't necessarily my thing. If you can see on the screen, I included a picture of some of the code that I was programming and this weird inexplicable black dot that appeared on screen. That's not supposed to be there. That was just some weird kind of hiccup that happened with my code. And I realized that maybe that was not the type of thing for me. The research itself was very interesting. I love physics and went on to do a degree in it. But personally, I realized from this experience, for all that I learned from it, that I wanted to be part of research that was more hands-on and lab focused. So with this in mind, when I came around to my next research opportunity, I applied for a Queen Elizabeth Scholarship internship in Malawi and ended up working at an internship there with the Center of Excellence for Water and Sanitation at Mzuzu University. So we did a number of different projects where I applied some of the skills that I had learned on the microbiology and cell biology part of my degree and the lab work that I had studied up to that point in school. So I did a number of different tests on water samples for coliforms and other physical data, and most notably, did a 3-week field project on the spread of cholera. And this was really interesting for me because we were working with a really diverse team. I was there as a group of lab technicians. So we were doing, once again, water testing for coliforms and kind of the microbiology aspect of it. But we were also there with surveyors who are tracking geospatial data to kind of track and chart the spread throughout the country and throughout the different regions. And also a number of public health analysts who are administering surveys to assess general knowledge and competencies for public health measures that could help prevent the spread of cholera. And what I found was that being at the hub of all of those different research interests was really interesting to me and really exciting, because all of these different groups were bringing something to the table that none of the other groups could. And all together, they were creating something that was far more interesting and far more cool, if I may say so myself, than the sum of their parts. So the biggest thing I learned from that experience research-wise is that I was really interested in interdisciplinary research that combined the approaches and the interests of a number of different fields and directed them towards one common goal. And it was especially interesting because cholera, dealing as it is with public health, did have a translational impact. So that became something that was important to me as well. So that brought me around to my third research opportunity, which is the one that I'm going to talk about most today. I did this in the form of an honors thesis, which is available to pretty much everyone in science at UNB. So it comes under different names, whether it's senior research project or honors research experience. There are comparable programs in engineering for senior design projects as well. I did my honors thesis initially with Dr. Janice Lawrence, but it was also supervised by Dr. Adrian Reyes Prieto. We'll talk about how that came to be about as I get into the research itself. And the focus of it was on the genetic analysis and detection of toxic cyanobacteria in the Woollistock. So without further ado, I would like to hop into my research experience with that now. So for some initial context, in 2018, there were a number of dog deaths on the Woollistock. So these dogs were grazing on the shores of the river around Carlton Park or the Northside Green, if you know where that is in Fredericton. And they presented with acute neurological symptoms prior to their death. So they started seizing, they became paralyzed, and eventually they asphyxiated. Now, not luckily, it was an extremely unfortunate situation, but the pet owners were very proactive and consulted the New Brunswick Provincial Veterinary Lab, who proposed that the cause of death could have been toxic cyanobacteria. Now, cyanobacteria, when you see it in the news, is often characterized as a bloom or a planktonic cyanobacteria. So something floating on the surface of the water that you're you're told to stay away from. But in this case, there was no planktonic cyanobacteria present, no blooms. However, there were drifting benthic mats found nearby. Now, benthic cyanobacteria, or benthic organisms of any kind, are those that reside relatively low in the water column. So I've included some pictures here of some cyanobacteria that are kind of lashed onto the surface of riverbeds. And you can see that they're not what you traditionally associate with cyanobacteria. They're not these bright blue or green colors. They're kind of brown and dirty looking. And they start at the beginning of the riverbed. Now, given that these are photosynthetic organisms, if they create a large enough aggregate, so we call these big aggregates here mats, then the buoyant forces from the photosynthesis and the oxygen that they produce can become sufficient for them to tear off from the bottom of their riverbed substrates and float downstream to different locations. Now, the other thing that is of concern is that when they are forming these large mats, these large aggregates, and there's high growth going on, there is the potential to produce various toxins, one of them being anatoxin A. So anatoxin A is also referred to as the very fast death factor, which sounds kind of goofy, but is actually kind of terrifying. So the way that it works, its mechanism of action, is it mimics the binding of the neurotransmitter acetylcholine at the neuromuscular junction. So typically what happens is an electrical impulse releases the neurotransmitter acetylcholine into the synaptic cleft, shown here, and the chemical binding of acetylcholine across the synaptic cleft initiates a muscle contraction on the other side. So typically the way this works is you get a tightly regulated system where you have the electrical impulse, the release of the neurotransmitter, and eventually the muscle contraction on the other side. And it's regulated by these degradative enzymes called acetylcholine esterases. So instead of having acetylcholine bound all the time and just constant muscle contraction or no muscle contraction at all, you have these enzymes come in and you break acetylcholine molecules down when they're not needed. However, there are no cellular enzymes that can degrade anatoxin A. So when anatoxin A binds, pretending to be acetylcholine, there's no way to remove it from the place where it's bound. So what results is uncontrollable muscle contraction, fatigue, and eventually death. And this is a really large concern because these systems are comparable not just in dogs but also in humans. So these toxins are certainly threatening if they reach sufficient concentrations, not just to dogs but really any vertebrates. So the anatoxin A biosynthetic pathway is fairly well characterized and well understood, and it's under the control of a suite of collinear genes called the ANA cluster. So there are a number of different genes associated with them. I'm not going to talk too much about them, but it's important to note that variation in this cluster can result in variations of the toxins produced. For example, ANA-J, which is located right here, is associated with a different toxin variant called dihydroanatoxin A. So we're not just getting one chemical product, we could get a different one as well. Now, we're of interest in this gene suite and in this pathway is ANA-C, which is the gene that codes for anatoxin A synthetase. That's the one that initiates the biosynthetic pathway and gets everything rolling. That's also the one that we ended up targeting in our genetic analysis with our primers. So to give you an idea of what we are going for with this project, we wanted to implement PCR-based methods to detect the ANA-C gene. And the reason for this is that we wanted to identify where the potential producers of anatoxin A were in the Woolastook. It's a very simple test and a very easy way to assess, is there a potential anatoxin A producer in this area? However, we wanted to take it one step further. We had information from 2018 research and samples of toxic cyanobacteria from that set of research. And we wanted to use genomic approaches to try and get a handle on the ANA cluster and what it looked like, what its structure was, which of those genes were present, what kind of variation did we see in all those different isolates that we'd obtained, and how could we use that information to predict the toxin chemistry that we were seeing and observing in our 2019 studies. All of this together would result in measures and information that could help monitor and manage risk for different watershed organizations, and in particular along the Woolastook. So the first section, there are two main sections of the research that I wanted to talk about, is the PCR detection of the NSC gene. So the way that it works is we started out with 8 sample sites that were chosen along the Woolastook. They're noted here. And mat or sediment samples were collected from each of these 8 sites from early June to late September. Once we obtained these sites and brought them in from the field, they were observed under microscopy just to try and give them a tentative ID. And samples were ground up using a tissue grinder and extracted using a commercial kit for their DNA. So basically we ground it up into little pieces, broke down some barriers and some cell walls and the like, and then used chemical procedures to further extract the DNA from our messy little containers. And once we had the DNA and it was nicely purified, we used PCR or polymerase chain reaction to amplify the NSC gene, which is the gene that initiates that biosynthetic pathway. So the presence of NSC is a strong indicator, not just that there's a high potential for them to be able to produce antitoxin A, but for the most part, if you see the NSC gene in general, you can generally conclude based on a number of studies that antitoxin A will be present. So if the gene is there, likely that organism in particular is producing antitoxin A. So what we found in this large table with lots of complicated numbers, or so it seems, is that in 127 of the 169 samples that we obtained were NSC positive. So there's at least one positive every week. And if you kind of just look generally speaking, most weeks are positive for most of the summer. So if you look a little bit more carefully, I have a few things that I want to highlight. Most weeks have a large number of positive samples. So in weeks 1, 4, 7, and 8, there are positive samples in every week that we ended up getting a mat sample from that region. And in 3, 4, and 6, in all but 1 week. Now, what we can also see is that there's a seasonal trend. So let's look at site 1, for example, or what's a better example? Probably site 5. So site 5, it's fairly consistently positive when we have more than one sample in the first few weeks, but then it cuts off a little later on towards September. So we were able to observe a seasonal trend of decreasing for the amount of positive samples that we saw in the field, getting close to the fall. And this has something to do with the temperatures. So cyanobacteria, when they're given ideal warm summer temperatures, are able to grow longer and to grow into these large aggregate forms called mats. The weather gets a little colder in the fall and there might not be the same conditions for them to grow that way. And so as a result, there's a little bit of a downtick with the amount of positive samples that we detected. Now, I've highlighted sites 2 and 5 because in comparison to some of the other sites, for example, 7 and 8, where there were positives in every week, these are a little spottier. There are more negative samples than there are compared to some of the other sites that we found. So we actually related this to the fact that these sites were related in back channels of the river. And not just back channels of the river, but also downstream from other watersheds, such as the Keswick River. So these different channels, which I can highlight here, could potentially be governed by different flow regimes, be subject to different nutrient content, and have different sediment loads than the main channel of the river. All of these environmental factors could contribute to why there is a significantly lower detection of antitoxin A and NSC genes in these regions of the river. So we wanted to observe that as well. It's not just whether or not it's present, there are environmental factors that contribute to its distribution as well. On the note of distribution, in addition to our main sampling sites, we also took a number of samples of interest that helped inform other parts of our study. So the first 2 that I would like to highlight are samples that were taken from the upper Woolastook near Heartland and Belle Bay, which is downstream of Fredericton. And we found drifting mat samples at both of these sites, and all the ones that we found there were positive. So this lends credence to the hypothesis that once these mat samples are big enough, there's a large enough population for them to photosynthesize, get sufficient buoyant forces to break off and float down the river, then that population is likely large enough as well to produce antitoxin. So these floating mats likely are toxic as well, which having this information from both upstream and downstream suggests that these mats are motile and also allows it to become very widespread throughout the river. We also took some drift samples that we found in the region of our regular sampling sites, all of which were positive, all of which contributed to that same hypothesis. And there was also, unfortunately, another dog death in 2019, and all 4 samples that we took from that region were found to be NSC positive as well. So saying not just that it's widespread, but also that it is in fact toxic, and it is the toxins that we're suspecting they are. So what can the PCR data tell us? First of all, that it's benthic cyanobacteria, not planktonic, so stuff that's in the form of a mat, not a bloom, that's producing anatoxin A and is in fact present and widespread in the Woolastook, which is consistent with the work that we collaborated with the Canadian Rivers Institute, who did a lot of observational and ecological studies around the same time and in tandem with our project. We found out that the distribution of NSC appears to be seasonal, but environmental factors may have an impact. So we did see a sharp, we did see a sharp decrease in the fall. However, we did see things like side channels and different flow regimes having different impacts on the amount of NSC that we saw in different regions. It should be noted as well that weather events, for example, a really blustery rainstorm that could shake things up and dislodge a lot of mats from their benthic substrates, can also have the same effect. So a little later on, after I was finished collecting data for my thesis, we did actually find another uptick in the amount of toxic mats that we saw, because they had been dislodged by a rather nasty fall storm. The other big takeaway is that these mats are motile, so they can move downstream in the river, and even if the source of the toxic cyanobacteria was, let's say, Heartland, that doesn't mean that it can't affect somewhere in Fredericton, because it can flow downstream and affect those areas as well. The second prong of this research was the genomic characterization of the anacluster. So the way this worked is we received a bunch of shotgun sequence data from the 2018 Woolastook isolates. So we had collected a bunch of cyanobacteria that had been NSC positive in 2018. We sent them away for mass sequencing. And then I took those sequences and assembled them into something called a metagenome in a program called SPADES. So it takes all of those different possible sequences and compiles them into a kind of composite form that can tell us a little bit more, not just about the individual sequence, but maybe about the community, what we're actually seeing in that area, in that region, genetically speaking. Once we had these metagenomes, you have to take all this cluttered information and kind of give a little bit of order and organization to that. And the process by which that's achieved is called gene annotation. So the basic idea is we used a program called Genius to highlight different potential open reading frames, different potential genes that were related to the anacluster, and identified them within these metagenomes, and then further confirmed them using TBLAST-X searches of known gene databases. Once we had all of those identified, we aligned these different clusters to compare and contrast how they were similar, how they were different, and to try and observe what kind of variations we could see genetically speaking that could explain variations in the toxin chemistry that we were exploring in other collaborations with other groups. So what we have here is the big alignment. It looks very confusing and mildly alarming when you look at it quickly, but I'll try and shed some light on it. The first thing to note is that of the 12 or so isolates that we assessed, 9 of them did have anaclusters. So all of them that we see here did have anaclusters, and all of them except for isolate G were actually producers of anatoxin A. So it's a little suspicious that we have an anacluster for isolate G, but no toxin detected in conjunction with that. But even more suspicious is that isolates A and B, which you'll notice are not indicated on this figure, did not have a cluster, but they also produced anatoxin A. So this suggests that there's a slightly more complicated system or control over anatoxin A production than we might think. The other thing that's worth noting is that every single one of these clusters that we identified does have ANA-J, which, if you'll remember, was one of those variants in the ANA cluster before that allows for the production of dihydroanatoxin, a different toxic variant which can have different, more acute toxic effects than anatoxin A, which, remember, is called very fast death factor on its own right. So not necessarily a fun thing to begin with. What we found is that if you compared all of these different clusters to each other, they were all reasonably similar, and they all had a great amount of similarity to a known cluster of Anabaena species 37, which is indicated by this kind of through line at these different areas of the genes. Those are different regions of similarity between all of these different isolates, and this particular isolate, which shared the most similarity to all of our test isolates. Now, what we found is that it looks very similar to Anabaena 37, but these last three genes, so Ana J, Ana I, and Ana A, were found on a different strand, on the opposite strand as they were in Anabaena species 37. So we decided to look into that and try and figure out if there was an answer within the genetics that could tell us why we had something so similar, but with such a small but potentially nuanced difference. And what we found is if you look at the structure of any of these isolates, you see that there's a set of known ANA genes, there's a fairly large gap in between, and then there's another set of ANA genes on the other side. But all of them have this somewhat characteristic gap in between them. When we looked at the different genes and different codes within that gap, we found a number of open reading frames and decided to investigate whether or not there were different protein coding genes within that area. And what we found is that there were two transposase genes present. Now, transposases are responsible for moving different genetic elements to other locations in the genome. So we concluded from this that these transposases could potentially be responsible for the inversion that we see. They could have been responsible for taking the genes that were present on one strand in the Anabaena cluster that we see and moving them to the opposite strand, as we see in all of the different clusters that we determined from isolates from the Woolstock. So what can the genomics data tell us? Well, first of all, 8 of 9 of the Woolstock isolates that we found containing the ANA cluster were anatoxin A producers. So the fact that the cluster is there certainly does suggest that anatoxin A production is going to be concurrent with that. We also found that there were 2 isolates without the ANA cluster that were identified as toxin producers. So this system of control could be a little more colorful than we necessarily think it is. The other thing that we noted was that all of the isolates have genes required to produce different toxin variants. So all of them possess that Ana J gene. All of them possessed the potential to produce dihydroanatoxin instead of just regular anatoxin A, which means that they could potentially be considered more severe, or at least more able to be creative in how they're going to affect and influence the communities around us. We also found, lastly, that all clusters were similar to Anabaena species 37, but notable differences. For example, Ana J, Ana A, and Ana I appeared on the opposite strand. But with a little digging, we discovered a number of different open reading frames in those gaps between those 2 large cluster segments, and think that there's probably a likely cause of transposition going on that moved those 3 genes from one strand to the other. So the big picture is that anatoxin A was widespread in the Woolstock in the summer of 2019, in the summer of 2020, just as a little spoiler for future things to come. And production of different toxic variants is possible. The big takeaway of this project, I think, was that PCR amplification of the ANA-C gene is a simple method for monitoring all of these different anatoxin A producers. So PCR is something that you do in your first year biology lab, but it's being used as a very simple technique to carry out very important work and very helpful work in terms of monitoring watersheds and protecting communities and protecting our animals. And lastly, understanding the ANA cluster allows us to make inferences about toxin producers. One thing that wasn't necessarily included in my thesis, but is part of an ongoing research project that we're carrying out, is we have collaborators in Halifax and collaborators in St. John who are carrying out different chemical tests to assess the amount of antitoxin A or different chemical products that are being produced. So we're able to use this genetic and genomic information to make inferences and to try and draw connections between the genetics that we're testing in our lab and the chemistry that they're analyzing in other labs. So you get a very large holistic picture of what's going on from the ecological side, the genetic side, and the chemical side as well. So with all that being said, I decided to wrap this whole thing up with some advice to some aspiring student researchers if you're interested in getting involved. My first piece of advice is to find out what research does and doesn't interest you. And the doesn't part, I think, is maybe more important than the does. So my first experience with research was in a fantastically interesting project. It was incredibly cool. I loved the physics that I was doing, but didn't really love the computation aspect of it. It wasn't really hands-on for me. So that was not something that I personally liked, and I didn't think that that is a bad experience. It was just a learning experience that showed me what I wanted to get into more. The second advice I have is to ask questions and make connections. So one of the reasons why I ended up with Dr. Lawrence is because I had her and made connections with her as a professor. I made connections with her when I was her PAL leader, but I also had friends who worked with her in her lab and were able to give me some information about how the vibe was, what the information and research was like. So I was able to make a really informed decision on whether or not I wanted to work with her. Obviously I did, and obviously it was a fantastic result for me. That wouldn't have been possible without those connections in the first place. Number 3 is to go for opportunities, even if they're long shots. So I applied for an NSERC USRA in my first year, knowing that it was a really competitive process and knowing that I might not get it. I wasn't really expecting to get it. I wasn't really expecting that research to get funded, but it turned out that it did. And as a result, that got my foot in the door and was able to open up a lot of doors for other future research opportunities just because I went for it, even if I didn't necessarily have the best chances of getting it on the first try. Number 4 is don't be afraid to learn or work with new people. So my project with Dr. Lawrence initially was just going to be genetic based, but having sat down with her and Dr. Reyes-Preto, we realized that there was much cooler information that could be shed on the project if we also added the genomics aspect as well. So I went into that project knowing absolutely nothing about bioinformatics or sequencing, alignment or metagenomes, whatever you want to call it. No experience whatsoever and a bit of a jadedness against computation, but decided that I was worth pursuing just because of the cool new insight that it shed on my project. So even though it was a little scary and even though I hadn't formally met with Dr. Reyes-Prieto, it ended up being a really great research relationship that taught me a lot of new things, not just in the lab, but also about collaborating on new projects and new techniques. And finally, follow the research where it takes you. It's not something that you can walk into and expect everything to be laid out black and white. It's not something where you know exactly where you're going right from the beginning, no matter how well designed your project is. Sometimes there are going to be things that are just surprising and you're going to have to follow them because you want to see where they go. And just like a good research project, that's kind of how my whole experience with undergraduate research has been. It's been a little all over the place. It's been very varied, but it's been very, very formative in my development as a scientist and I hope as a person. And I hope that you can take some of the same steps that I did and get some of the same takeaways as well. So last thing, really quickly, just want to acknowledge everyone who contributed to my thesis. Dr. Janice Lawrence, Dr. Adrian Reyes-Prieto, who supervised my thesis, Dr. Sean McClellan, and Dr. Cheryl Patton, who were on the panel for my defense, Kristen Hawks and Sweta Jayachandran, who were collaborators in the lab, Dr. Megan Bruce and the field sampling team from the Canadian Rivers Institute, our collaborators in Halifax and at Dalhousie for sequencing and toxin chemical data. And of course, thank you to the Atlantic Student Research Journal for having me come and speak today, and all of the SciSpire attendees, panelists, and speakers. Whew. Questions?
Thank you for the talk. That was so amazing. If anybody has a question, again, just raise your hand and I'll call on you.
I have a question, Lauren, but I don't think I can actually raise my hand because I'm a host, and there's no button there for me to raise my hand. Also, just to let everyone know, you can raise your hand by clicking on, I think, participants, and then you can raise your hand through that. But I have a question for you, Jake. So you mentioned a lot about bioinformatics,
Yeah.
and I was just wondering if you think this is like a fast-paced kind of developing field, or is it something that's been around for a long time?
I think both. It's certainly not a new thing, and I don't think I was doing anything super complicated or super cutting edge with the bioinformatics or the sequencing that I was applying. It's a fairly common tool in a lot of biochemistry research. One thing that is, I guess, a lot more exciting about it is that even though the applications that I applied were fairly rudimentary, people who are quite savvy with it can get really cool results out of it. So whereas I just looked for the NSC genes and the ANA clusters within all these different isolates, different bioinformaticists on the project who are working on this same project have used their skills to identify full-on pathways, full-on metabolic pathways and different genetic systems of control that are much more complicated, much more sophisticated than I could. So it's being able to amass and direct this incredible amount of information, incredible amount of data, and to find these patterns in it. And those, some of the patterns that have been highlighted through the savvier bioinformaticists among us have actually gone on to guide the project after what my thesis completed into a new direction. So definitely very much developing, but I don't know that I can necessarily speak to it very much beyond what I did in my thesis.
Thanks a lot, Jake.
Dr. De Verdemacher has a question.
Okay.
We can't hear you right now. No.
It doesn't seem to be working.
Yeah, your mic doesn't seem to be working.
Maybe if you go in the bottom left corner and say join with audio, there should be like a button down there.
Maybe like this?
Oh, there we go.
Yay.
But you don't see my video, do you?
No, we see your video.
You see, okay, so you saw me struggling. Well, thanks Jake for sharing your experience. I have a question more like not about your research specific, but how would you say that being a physicist has helped, was of benefit, or worked against you in this research project? So both sides, like where do you think it was an advantage? Where do you saw it as being a disadvantage?
So I definitely think in general, a knowledge of the physical world and how physics works is certainly advantageous in any branch of science. Maybe that's just personal bias, but that's how I feel. The project itself was not particularly physicsy, so there wasn't a whole lot of physics that came up in the project. However, it was handy because we use a lot of tools that rely on physics. So if you are doing a PCR and then running those amplified genes out on a gel, that process is called electrophoresis, is based on, electric currents and different, excuse me, different processes that I learned about separately and perhaps more theoretically in physics. It's also, I think, really relevant to point out that whatever my personal feelings about computation, that is something that is very important in a lot of physical studies, and it's something that I was exposed to a lot throughout my physics degree. So the fact that I had to take courses where we needed to find numerical solutions or use numerical methods to solve equations or to solve problems or to model systems throughout my undergraduate degree certainly made me a little less hesitant to approach different computational tools in biology. They're obviously doing very different things, but the fact that there was some computational literacy from my degree certainly helped and made me a little more braver to pursue that angle of the project.
Awesome. you have a question from an anonymous attendee who
Okay.
asks, for first-year students, how can they start their research journey? Who can we talk to and connect with to learn more?
So in general, I would say NSERC is one option to look for, just because it's not restricted by age. There are fairly stringent GPA cutoffs, so it's quite competitive and really kind of depends on the year for how they assess who gets the awards. But a lot of profs just have, in some years, just money kicking around that they might have, or extra funding to fund a student. So for example, my research with Dr. Lawrence, I worked with her the summer before I did my thesis, wasn't funded on NSERC. It was funded through, I think, through an environmental trust fund, not an environmental trust fund, but a research fund from a different environmental group, not NSERC. So there was money kicking around from different sources, funding kicking around from different sources. In terms of how to make those connections, I definitely was able to be more comfortable to have open and frank conversations with Dr. Lawrence about the different research opportunities that she had, given that I had such a good relationship with her as her student in a couple of classes and as her PAL student. But when I approached Dr. Hussein in first year, I had never had a class with him at all. In fact, I think I considered applying. I reached out to 3 different people to do summer research that year, and I had never met any of them. But I just put myself out there and asked if they had anything going on. So really, it's just putting yourself out there and trying to make those connections. You don't have to have them right in front of you. If you're willing to put the effort in, then there's a good chance that you might get something out of it. At the very least, you'll have a contact who you can build a relationship with in the future.
Awesome. Does anybody have any more questions? You can use the question and answer. Jackson?
Hey, Jake, that was a really great presentation. Thank you for joining us today and sharing. You emphasized collaboration in both your QES research project and your honors project.
Mm-hmm.
Could you speak to maybe the increasing importance of collaboration in scientific research?
For sure. So I think my big thing, and I think a lot of people would agree, is that there's a huge tendency to try and compartmentalize different branches of science, to say that biology research is just biology, or physics research is just physics, or chemistry is just chemistry. When in reality, the lines between all of these different fields are a lot more sinuous than that. And there's a lot more overlap than we think. And even if there's not direct overlap between, say, the ecological impacts of cyanobacteria present in rivers and watersheds, there definitely is an overlap in the tools and the approaches that we can use from chemistry or physics. So in this one project that's nominally a biology project, we have toxin chemistry analysis going on, we've got mass spectrometry going on, we've got biological genetic data going on, all being applied towards the same project. And not one person can necessarily master all of those things. So we were able to get such diverse and informative products out of our research because we were collaborating effectively with 3 or 4 different teams, all of whom brought something different to the table, and all of whom enhanced our research and made it a little more interesting and a little more deep and a little more nuanced than any one of us could have brought individually.
Awesome. I think we have time for one more question if anybody has anything last minute they want to ask. All right. Well, thank you, Jake.
Oh, I had a question, sorry, Lauren.
Oh, okay.
So when you went to Malawi, did you find the atmosphere regarding research different because due to like different cultural differences, or just like the way that research is conducted in Malawi?
There was definitely some adaptation that needed to be done for cultural norms and different modes of communication, just in terms of being in a different place that wasn't in Canada, not just in research either, just as a general rule. The thing that I think was most different about how research was conducted, because the projects were still kind of laid out and well designed and well informed and that sort of thing. So in that sense, there wasn't a whole lot of difference. But because of the different locations that we were working in, we didn't necessarily have the same equipment or technology. So for example, I would have done more complicated, or at least the equipment we would have used would have been fancier in the third year microbiology lab that I took at UNB than a lot of the field equipment that we were using in Malawi. That's not to say that we weren't able to get good information and good results, but it was adapting to tools that I hadn't used before and tools that were very different from those being used in Canada. So even though the end results were the same, getting there both communication wise and equipment and in practice wise was very different.
Awesome. Thank you very much for your presentation.
Thank you for having me.
Now it's time to move to our second speaker. We have Dr. Stijn de Berdemaker, who is an associate professor in the Department of Chemistry at the University of New Brunswick. As Canada Research Chair in Theoretical Chemistry, Dr. de Berdemaker works on translating ideas from mathematical physics, theoretical physics, and machine learning into meaningful chemical descriptors. So feel free to come on, Dr. de Berdemaker.
So can you hear me again?
Yep, we can hear you.
Wonderful. Okay, so let's get share the screen, I suppose. Is this visible?
We can't see your screen, but it says that you're screen sharing.
Can you see something? Can you see it?
No, not yet.
Well, it is on. I put it on full screen. I just see your.
We just see that it says that you've started screen sharing. Could you try going to your screen?
So maybe I need to give it some, but for some reason, I'm just gonna, I think I'm gonna log out and I'm gonna log in again. Is that fine?
I think maybe if you when you go to share your screen,
yeah,
there's like
the problem is now that for some reason I don't seem to be getting back to that screen. Something has blocked. I think I'm gonna have to log out and in again, but I don't know how to do that even. Give me a moment.
Oh, there we go. We can see it, I think.
See anything? Ah.
We see your screen, kind of.
Is it coming up?
I can only see like I don't know what it is. It's like your document viewer or something.
Okay. What if I like? Do you see something now?
Yeah, it's working now.
Aha. Okay. It looks like okay. I think I understand it, but that doesn't matter how I. So how do I navigate this thing? Yeah. Okay. Weird. So yeah. Okay. You are able to see the presentation?
We certainly
Yeah.
see it, Dan.
Okay. Yeah. I for some reason I'm not able to maximize the screen. I'm gonna try a new share. Okay. Gonna. Ah. Okay. 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 So this. Do you see the slides or not? Question? Can you hear me?
We don't see the slides right now.
So I have to put it in the other one, and then. Can you see it now?
No.
You can't.
No, I can just see you now.
This one, share. Can you see it?
Yeah.
Yep, there it is.
Ah, okay. So we're good to go?
Yep.
You can see me?
Yep.
Okay, wonderful. So I'm just going to put myself a bit on a timer because when I'm talking, sometimes I keep talking. Okay, wonderful. Good. Good. Okay, so well, thanks a lot for giving me the opportunity to talk a little bit about our research. When I received the question, then you asked me to talk about a project that I started 2 years ago, but it was very hard for me to choose one because I'm in real theoretical chemistry, I have to take like a very long build up to get to the place where I can talk about the project that we started 2 years ago. So instead of that, I actually decided to talk a little bit more about my journey into where I'm now. Okay. So the title is Lewis pairs in chemistry, from nuclear physics to quantum chemistry. So a little bit in the maybe there's a theme here, but I also started out as a physicist. It took me a little longer than Jake to figure out that maybe that was not exactly the place where I wanted to be, but it's very similar. So yeah, I'm in the Department of Chemistry at UMB here. You can also check out some of the stuff we do at the website, and I'm fairly active on Twitter. So here, this is me in full. I think that was the last time I put on a costume. Presenting, or when I was presented as a tier 2 CRC, Canada Research Chair in theoretical chemistry here at UMB. My name is Stan. It's pronounced for Anglophones more something like Stain. For the Francophones here, it would be Stan with the accent grave. If you mess it up, that's not a big deal. So like I said, I started off my my degree in nuclear structure physics at Ghent University. So I'm Belgian by origin, let's say. I sort of like wandered off in all sorts of directions, and I ended up in theoretical chemistry. So recent research projects are in quantum chemistry and mathematical physics, machine learning, and we took up some projects in quantum computing as well. Okay, so I'm going to talk a little bit about the road that I took, the road to chemistry, and more in particular, the road to Geminos. Okay, and I'm going to talk about what Geminos are. Okay, so yeah, quantum is a bit of, let's say, has a bit of an aura in the sense that it's weird, right? And sometimes it's like, yeah, it's weirdness for weirdness sake, but that's not true. We do have a longer purpose, like a long-term purpose, if you wish, because there are challenges in our world, right? And some of the challenges are are environmental issues, right? It's undeniable. Like climate change is not a vision; it's a fact. Okay, so we'll have to deal with that, or we have to make sure that we can mitigate sufficiently early in time. But there are other issues, right? Our population is aging, so it's gonna, as we grow older in general, and there are more of us, healthcare is gonna put a big stress on our society. So there are many ways you can deal with it, and one way you can deal with these changing societal parameters or variables are by means of science. Okay, and this is our angle. So if you look at those two objects that I put on my slides here, the one on the left, or maybe let's start with the one on the right, that is a micro, let's say even a nanomagnet. Okay, so as we rely more and more on computational resources for everyday lives, building computers, building technology is putting a lot of stress on environmental resources, right? So getting good materials is one of the ways how we can work towards a better environmental thing, but by better environmental environment. So what you see there on the right is a nanomagnet. It's just a couple of atoms, right, containing some iron atoms, and they can behave as a magnet that can go into your hard drive, that can run your tomorrow's computer. On the left side, what you see is a protein. Okay, it's called amyloid beta. I'm not an expert on amyloid beta. What I know is that when patients with Alzheimer disease, when they get a post-mortem dissection, then they see that these amyloid betas, there's all sorts of plaquettes formed in our brain cells, right? So that is definitely not, well, these plaquettes, they build up and they make even spikes and they pierce through the cell membrane. So that is definitely not a way how these proteins are supposed to behave. So getting a good understanding of what these proteins are doing in the cells is going to be of major importance to understand that illness better. Okay, now, yeah, that's fine. It's like two completely different materials or different forms of matter. Like one is biological, it's proteins. The other one is material science, it's iron. So how are they connected with each other? Well, the answer is pretty simple. They're connected by means of quantum mechanics. Okay, the world at which these systems behave is the world of quantum mechanics. And so we need to get a very good understanding of physics in this way, or quantum physics, in order to understand how these materials or how these systems are behaving and how they are made. Now that's cool because quantum's cool, but it also poses a lot of issues. And it's interesting because quantum mechanics or quantum physics came about in the 1920s, okay, at the end of the 1920s. And immediately this guy, Paul Dirac, is one of the founding fathers of quantum, he immediately saw a problem, okay? So in one of his early papers, he wrote down this sentence I'm going to read with you. He says the fundamental laws necessary for the mathematical treatment of large parts of physics and the whole of chemistry are thus fully known. All the laws were known. So they know about Coulomb interactions, they knew about energy conservation and all that stuff. So everything was known. And he says, and the difficulty lies only, remark only, in the fact that application of these laws leads to equations that are too complex to be solved. So this guy saw trouble. Okay, and there was trouble, and that trouble is what we've been working on the past century almost to resolve that. So okay, so what's the problem with quantum? The problem is here. Everybody, or I think you might have heard about Schrödinger's cat. Okay, so Schrödinger's cat, what's Schrödinger's cat? Schrödinger's cat is like a Gedanken experiment, a thought experiment by Schrödinger and Einstein, I think, that said, okay, everything in nature, or all quantum particles, they can be at multiple configurations or multiple states of reality at the same time. Okay, so they form linear superpositions, I said the word, of different realities or different states of matter. Okay. And yeah, so these guys, they ridiculed it, said, okay, now if we put a cat in a box and we don't know what the cat is doing, and we put him together with a radioactive quantum particle that may decay or not, then we'll never know whether the cat is going to be dead or alive, right? If that quantum particle, that little amount of radiation releases a poison in that box. We'll never know. So the cat is going to be dead and alive at the same time until we open the box. Now, that was meant to ridicule the quantum theory, but it turned out that it was actually pretty accurate. So all particles can be at multiple states of reality at the same time. And it's only when we measure it that it'll collapse to one that we can observe, but that's a completely different story. Now, how does that relate to the issues that Dirac foresee and that we are trying to solve? Well, I think I can illustrate it by means of this cartoon. So let's say you want to build a molecule from quantum mechanical purposes, from quantum mechanical principles, ab initio. Then how do you start with that? So first thing you do is molecules, they consist of nuclei, positively charged nuclei and electrons that circle around them. So the first thing you do is you put your heavy nuclei somewhere in space. Okay, so that's the first thing you do. Now, then you're going to put some electrons around it. How are you going to do that? Well, basically what I'm going to do, I'm going to discretize space and I'm just going to create boxes. Okay, so I'm going to just make boxes and I can put my electrons in some of these boxes. Okay, so here are my electrons. There they are. And you can see I put them in boxes that sort of make sense, right? So they're around the charges, some are between the charges, and that's a perfectly acceptable configuration, okay? Now, this one is also actually a pretty good configuration. I mean, it's pretty reasonable, right? And you can go on like that, right? So you can also make that. This one is probably not so likely, huh? This one is probably not going to occur, but this is what you. So this is what you get. So you get a whole bunch, a whole huge collection of all possible realities that can happen. And quantum mechanics now tells us that we have to build, if we want to build a quantum state or a quantum description of the system, then we have to take into account all these configurations. We call them Slater determinants. Okay, this is a technical term. And what we do is we make a sum. So there's a sum. So psi is a quantum state. We make a sum over all possible quantum configurations. Okay, classical configurations, because there you can think of like really a classical configuration distributing electrons into boxes. Now, then the problem, and this is the problem, is how many boxes, how many ways of distributing these electrons over the boxes are there? Turns out this is an exponential problem. And this is called the dimension of the Hilbert space. That's also a technical term. That doesn't really matter how it's called. The problem is that it scales exponential. And while we heard a lot of talk in the past month of how fast exponential can go, And this is really what's the big problem, because an exponential of a big number is not a big number, it's a huge number. So in order to illustrate that, I put here for all sorts of molecules. So there is H2, so the hydrogen molecule, there's methane, there's just the iron atom, I put benzene there, I put my favorite molecule, I'm still a physicist, caffeine also there. And basically what I do, I plot the amount of terms that you need as a function of how many boxes you allow for, right? Okay, so L is the number of boxes that you can have, and they all have a fixed number of electrons. So like methane has 10 electrons, iron has 26, caffeine has 102. And then the many ways, the different ways, how many different ways you can redistribute these electrons in this number of boxes. As you can see, it grows with the number of boxes. So the more boxes you allow for, the more configurations you can get. Now this is good, except that if you notice the scale on the y-axis, it's a logarithmic scale, right? And for your reference in red, what I put there is the number of the total amount of numbers you can store, you can work with in 1 gigabyte RAM laptop computer. Okay, I mean, maybe you have 10 gigabytes, but that means that I have to shift my red line just one notch up. So that is not going to make a lot of difference there, right? So it's a little bit of give you an idea of the ballpark. That hydrogen, for instance, a hydrogen molecule, I can do perfectly well. And we love hydrogen because we actually can almost do it on the back of an envelope. So as a physicist, we love hydrogen. But as soon as you run into something which is even a little bit more difficult, like methane, or let's say iron, the stuff that goes into that micromagnet, you immediately run out the capacity of your computer. Note that Moore's law, like the doubling of the number of chips on number of transistors on a chip every 1.5 years, is not going to help you either, because it's not going to put you. It's going to bring this red line a little notch up, but not too far. So there's definitely an issue with this prescription. It's we cannot go that way. It's just a losing game. We cannot go that way. We have to do something else. So what you do as a physicist, then you sit down, you watch the squirrels playing in your garden, as you are doing right now, and you think of what are the most important parts of the molecule. So what is the most important, right? So you have an exponential amount of a gazillion amount of configurations. Some of them are more important than other ones. Now, if you're a chemist, then you know that molecules bind together by means of Lewis bonds, by means of Lewis pairs. Okay, so I have a cartoon here of a hydrogen molecule. So you have two hydrogens, one on the left, one on the right. And in the middle, what you have is you make bonding orbitals, okay, and you make anti-bonding orbitals. Obviously, when you want a molecule to bond, then you want 2 electrons in this bonding orbital down below, and that is what drives your that's the most important one. Now, when you are pulling these two apart, so when you're doing the chemistry of this thing, when you're going into a reaction process, then this other one, this anti-bonding one, is going to become much more important, because you're stretching the bonds, you're breaking the bond, the anti-bonding is going to become more important. So you also have to include that one. But basically, in the end, what it is a resonance between this bonding and this anti-bonding, and those two are the most important ones. Okay, so and you realize that. So if you then do a calculation, okay, very on the back of the envelope of the system, and you only take these two configurations into account, you end up with this green curve that I put up here, which is the correct curve. Whereas other methods where you don't take those into account, where you just take one of the configurations, then you end up with descriptions that will give you an energy, a dissociation energy that's going to be twice as wrong as what you would have wanted. Okay. Now you can extend that. So this was a hydrogen molecule. Basically, every chemist knows that if you draw a molecule, you draw these fancy lines between them. Here's my favorite molecule again. This is caffeine. You draw these lines there between all your atoms. And those are the bonds. Okay. Now, what I want to have, I want to have a very simple, extremely simple description of that. I don't want to go through that exponential scaling. I just want to have the most important ones. So I want to have some sort of like a mean field description for all these pairs. How do I do that? I do that by building these so-called geminal states. Okay, so what is a geminal state? This is a little technical thing, but it's just a product. So a product I can work with, that's very simple, of stuff that is like a linear position, linear superposition of all these possible pair states, and bonding and anti-bonding, like I talked about before. Now, this is not new. I mean, my group, my collaboration, we did not invent this. This has been around for a long time, since the 1950s, proposed by Hurley, Leonard Jones, for instance, the guy from the Potentials. Okay, so he was also guilty of proposing that idea, which was awesome. It really describes well what it should do. The only thing is, and here comes at the whole, this is the whole, basically the whole gist of my research program, is that it turned out that these objects were computationally intractable. Okay, so still you made a simplification was sort of like the most important ones, and still you ended up with something that you simply could not work with. Okay, so this project was abandoned for other approaches that I'm not going to talk about too much. So the end of my story. So after, you'll see that we're going to work towards a theory that will allow us to compute these geminals. But the way how I got there, I'm going to talk about a little bit more now, because it gives you a bit of an insight of how these ideas came about. So like I said, I have a background in nuclear structure physics. Okay, so here is the hierarchy of matter. I'm sure you've seen that many times before. So on the upper left, there's a crystal about, let's say, a nanocrystal, 10 nano-sized. If you zoom into one of the atoms, you get into the angstrom size, and then you can see the electrons. And a tiny little speck, it's a little bit exaggerated there, of the nucleus that's sitting in between, because it has a femtometer size. Right? And a nucleus consists of protons and neutrons. Okay, and each proton and neutron consists themselves is made up of quarks. Now, my ballpark when I was doing my PhD was trying to get the quantum structure of the protons and the neutrons together into this in the atomic nucleus. Now, so yeah, it consists of protons and neutrons, but it had extreme issues. Protons and neutrons are composite particles. It means they're not unique point particles. They're not fundamental particles. They're made of quarks, right? And they interact by means of the strong interaction, which is a fundamental interaction, but not a fundamental interaction on the proton-neutron level, but on a deeper level on the quark levels. So basically, after working a couple of months in nuclear physics, you realize that they should have changed that name into unclear physics. Something you come to realize fairly easy. So you immediately learn that you have to look for the most important degrees of freedom. Now, at that point, experiment helps you. So because if you're looking at what I call the neutron separation energy. What is the neutron separation energy? The neutron, the name speaks for itself, is the energy that I need to take a neutron out of the nucleus. Okay, so it's the binding energy of an atom, a nucleus with n neutrons, minus the binding energy of one with n minus one. Okay, so if you plot that as a function of n, so this energy, this is in the MEV, then you see the staggering, right? This odd even effect. So it means that it is much cheaper to take out a neutron when it does not have a friend than when it does have one, right? So they make best friends forever, and it's extremely hard to separate. So there's a pairing effect. Okay, so I started working on that. And fairly quickly, and this is no secret, you learn that these pairing effects lead to a phenomenon that's called nuclear superfluidity. Superfluid is like a quantum fluid. It has many strange properties. But you learn that this effect was basically also known in superconductors. Okay, so a superconductor is a system where the resistance completely drops to zero. So electrons, they can just flow through the material at zero cost, zero resistance. It's just like free transportation of electrons, right? Now, Bardeen, Cooper, and Schrieffer in the 50s have realized so that the effect was known since, I think, 1907, pretty early. And it was only explained 50 years later, also by this pairing effect. So electrons tend to pair together to cause this superconductivity effect. So there is a correspondence there. So there's a similarity between these two systems. Now, at that point, I had graduated and I decided to move to Canada to work at the University of Toronto with Professor David Rowe, who unfortunately passed away a couple of months ago. He was a mathematical physicist, okay? And one thing I learned from him is that when science is universal, when you see the same phenomena popping up in different places, then mathematics is the common tongue. So then the language you have to speak is mathematics. Okay. So I learned a lot about, during my postdoc, about what I now call Richardson-Godin integrability. Okay, so there was a concept, a mathematical concept that I had to learn. And there you can see on the right-hand side, basically what it meant: buying a notepad, buying some good markers, some good Sharpies, and do a lot of calculations because it's math. Okay, so now let me explain a little bit further what this term integrability means. I'm going to start with a very loose definition. A quantum system is integrable when it supports as many symmetries as degrees of freedom in the system. So why is this so? Why is this so important? These symmetries. If you have a symmetry, then something is conserved. Okay, so we know that from physics. So once you have, let's say, rotational symmetry, then we know that the system is going to stay the same if I rotate it a little bit. The same with this beautiful snowflake here. If I rotate it over 60 degrees, I know that the system is gonna remain the same. Okay, so we try to find that helps us a little bit. Okay, so that it reduces the complexity of the system. Oh, I'm running slow. So that helps us a lot. Now, in terms of mathematics, so one of the main results that we got was that for these superconductors, we could write down a mean field, one of these product wave functions that I talked to, this product quantum state, so a product of a linear combination of states, provided that the coefficients, the x coefficients there, they fulfilled a special set of equations that are presented there at the bottom. Now, okay, that's nice, that's fine, but who cares? Well, we care, because if you look at the complexity of these equations, you see that the number of equations scales with n, and n is the number of electrons. So now I have something that is as difficult as the number of electrons and no longer this exponential scaling. So we knew we were in business with this sort of stuff. Now it turned out that these equations were extremely difficult to solve numerically. So my first part of my postdoc, I spent a lot of time trying to actually solve these equations. And then after a couple of years, let's say, I finally managed to do so. And as a demonstration how this sort of stuff works, you can, I have a little, animation of electrons that I'm putting into an optical lattice. So the blue is like the wells of an optical system. Now I'm putting electrons in there, and I can tune the interaction between the electrons. And then the red dots are sort of like the the electron energies, and they have a complex and a real part, and how they behave with respect to the wells, that are the blue dots. So you can do these simulations, and what we gather from these simulations are how quantum these electrons are. Okay, that's nice. So we had a numerical handle, so we could do computation. So then we were obviously interested more into applications. So where can we take this whole business? Turns out we could, because the common language was mathematics, this could sort of reach out to many different fields. Okay, so one of them was, for instance, we used this to look at time dynamics in diamond vacancies. Diamond vacancies, well, nitrogen vacancy centers in diamond. So you have a perfect diamond structure, and you make a defect there. So you put in a nitrogen and a vacancy together, and that can act as a qubit for a quantum computer. So that was what we worked on, was a perfect model to work on those things. So we investigated that. We looked at nanograins, superconducting nanograins, as a function of the size. That's the panel in the middle. And we also looked at some exotic P-wave superconductors, some really bizarre superconductors that may push the critical temperature to room temperature. Okay, I didn't do this work alone, right? So many of these applications I did in collaboration with Dimitri van Eck, Pieter Klaes, and Mario van Raamdonk. So these three people, they're all physicists, okay? And we were very much interested in all these sort of applications. And one of our applications was rewarded, as to say, with an interview in the Journal of Physics, Mathematical and Theoretical. So that was a nice day. So you start from the physicist, and then you add some chemists. Okay. So these are all the people that I've worked over to bridge the road, or bridge the river towards chemistry. So the people on the left were the physicists. Now, if you add some chemists, more in particular Paul Ayers from Department of Chemistry at McMaster University and his gang, then you can reach out to even more, let's say, applications. Because when I was doing this sort of work, I met Paul Ayers, this chemist from McMaster, and he said, hey, you know what, these objects, these quantum states you're working with, we know what they are. They're Geminals, but you can't do anything with them, he said. Yeah, but I said, we can do stuff with them. And hey, that's interesting. So we started working together, and we realized that we could design a whole bunch of new Geminals, right? That could describe this bonding, anti-bonding resonances fairly well. So you see on the right-hand side, it's like our proof of principle calculation, where the black curve is what it's supposed to do, the energy, the dissociation energy as a function of how far the hydrogen, in this case, are apart, with respect to some other methods, like the green and the blue, and they all behave ill, but we had our new method that was doing fairly well at which was also important, computationally cheap costs. So we had we thought we were definitely in business, right? Simply by using another concept from physics in chemistry, we were able to do something better. Obviously, nothing is perfect, which brings me basically to the project of the last 2 years. If you do that, if you apply this thing on benzene, you would see that you artificially break. So everybody knows, I hope, well, at least have seen chemistry of benzene, where you have these beautiful hexagons, perfectly symmetric. Well, it turns out if we apply our theory on this, it breaks this 6-fold symmetry. So that was definitely something which is not a good thing. So we had to open up our methods, allow not only for paired excitations. So what you see here is a diagram in which the upper echelon gives you only paired excitations, our Geminals. We had to break up some of these pairs. And so V=2 means I have broken up one pair, got 2 extra particles that are not paired. V=4, I've broken up 2 pairs, etc., etc. So I increasingly allow for these broken pairs, and then I see, as a matter of fact, that if I look at the energy profile as a function of the angle, it should be minimal at 60 degrees. If I start at the seniority 0, the v equals 0, which is the blue curve, it is more a minimum around 57. But if I include up to 6 broken pairs, 3 broken pairs, seniority 6, then I'm able to restore the system again, which is not unsurprising because I have 6 atoms, right? So 6 lone rangers is not too crazy. Okay, so this is sort of like an overview of how we got there. Obviously, there is a lot of stuff that needs to be done, and that's is the exciting thing about research. You do something, you solve a problem, or you get somewhere, and you immediately see so many other branches that you can explore. Right? So next things is going to be is we're going to probe them in a time-dependent way. Gonna get some shoot some electrons at them, shoot some light at them, see how they behave. This is one thing. Another way that we can another direction we see is that we want to move this up to more extended systems like vitamin B. Recently there's been an application with our method in this paper that I put the reference below on like the core of vitamin B12. So that is that was very exciting to learn about. And in the end, we also want to put our terminals on a quantum computer because, like we all know, the future is quantum. Okay, I'm going to wrap it up here. I'm extremely grateful for people that keep us off the streets, that recognize that what we are doing is worth exploring, is worthwhile working on. Those are the FWO in Belgium, who has funded lots of my international travel that brought me to Canada, and obviously to the Canadian authorities that are very enthusiastic about this research and support it. So in conclusions, sometimes your path is not linear. So in this case, I went over nuclear, over mathematical physics, and ended up into quantum chemistry, and all on the same team. Electrons are mainly paired, so use Gemmas. Well, and with this, I conclude my presentation. Thank you for your attention.
Thank you very much. Does anybody have any questions? I think we have time for 1 or 2.
I have a question for you, Dr. Bitterberger.
Yeah, speak up loud. It's hard to hear you. Yeah.
Oh, sorry. Can you hear me now?
Yeah, I can hear you perfect.
Okay. So my question is, on one of your equations there, you mentioned that the dim that I mentioned is H.
Yes.
What does H stand for in that?
Oh, yeah. Oh, it's the Hilbert space. It's a very technical thing. So the Hilbert space, it's like quantum space. So we are used to thinking in Euclidean space, right? 3 dimensions, X, Y, and Z. So 3 dimensions. Now, if you're thinking in terms of quantum space, every time you add another cat, let's say, in your box, so you have your Schrödinger's box, right?
Right.
Every time you're adding another cat in your box, you're basically multiplying your space, right? So
Because
you
you
start
don't.
with 1 cat, you have 2 dimensions, okay? Then you add another cat, you suddenly have 4 dimensions. So H is 4-dimensional. If you add another cat, you're going to end up with something that's 8-dimensional, 16, 32, 64, 128. I mean, I don't have to explain you how it explodes, right? Because this is inflection rates. So H is the Hilbert space. It basically is a space of all possible boxes all sorts of configurations that you can make. Maybe. Does it answer your question, or am I?
No, that makes sense to me. Thanks.
So this is the space of all possible of these, because I can make a linear combination of them. Okay. That is one.
Okay.
Yeah. It's like a vector in 2 dimensions, dead and alive.
Melina Paz has a question.
Yeah, so I just have a small question, and I was just wondering how your experience with such different fields of research impacted your experience as a researcher and just, yeah, your
Okay,
overall
that's a
experience.
very good question. Obviously, when you're in different fields, or you're coming from different fields, you obviously think about how am I, first of all, how am I different? Eh? You contemplate the diversity of the field a little bit more, and you also think like, where's my position? Okay, how can I contribute here? And in order to do that, you need to listen very carefully to what people are saying. And because you're coming from from one angle to a problem, right? So you come in and you have your experience and your background, and you're looking one way into a problem. And you also have a certain language that you use, right? Some like Hilbert space. I'll talk about Hilbert space, blah blah blah. But then a chemist or a biologist may ask me like, hey, wait a second, I'm interested in proteins, but what is this Hilbert space, right? And ask me this question again. Ask me this question, and then basically you explain it then, and then you better understand what each other's, how, yeah, how why there are the problems, that why they are there, right? So what you're basically working on and how you can contribute to that. Yeah, I'm a little bit vague here. I should be able to phrase this a little bit better. I think the main message here is that you have to be open to listening to what other people's problems are, and maybe not necessarily try to solve it immediately, but more take a step back and say, okay, so this is how they approach the problem. How would I approach the problem? And then see whether your approach can actually help the other. Right? And be open to that. Right? You also have to be open that somebody comes in and says, hey, what you are working on is something that that we know how to deal with, or we do it this way, right? And see how you can learn from that, and maybe try to solve some of your own problems by that. So this has been communication, basically. I think that is the most important part in interdisciplinary research. It's also very difficult because difficult. It takes a long time before you're on the same page. Because you step in and people say, oh, we have this problem here. And you look at it from your own angle, but people have been working on that for a long time. You're not just going to walk in and solve those problems. If that was just easy as that, people would have solved that already so long ago, right? So you can contribute. You need to identify how you can contribute best. Use your strengths without overusing them. Make sense?
Yes, thank you. That is awesome.
All right, so.
Oh yeah, and maybe another thing, don't be afraid to ask questions. This is one of the wonderful things that I think about, that I consider of interdisciplinary research. There is a potential to learn so many things. You can be a kid again. You walk into the room and there's this new toys and you don't know what they're for, right? You know you want to play, and you ask tons of questions. Nobody's going to say you're stupid for asking these questions, because you come from a totally different background. So there's an opportunity to learn so many things. If that was not a commercial for interdisciplinary research, I don't know.
Thank you, Dr. de Berdemacher.
My pleasure.
Our third speaker is Melina Paz. She's an undergraduate student at UNB, finishing her last year of her biology chemistry program.
Yeah, so hi everyone. My name is Melina Paz, as you guys just heard. And I'll just be sharing a little bit about the work I was doing this summer with my supervisor, Dr. Sarah Eisler. I'll just start with sharing my screen real quick to show you guys my presentation. Let's see. Can everyone hear me all right?
Yeah, we can hear you.
Okay, awesome. So I'll be sharing. Can everyone see my presentation?
Yep, sure can.
Okay, awesome. So like I said before, I'll just be sharing a little bit about my research with Dr. Sarah Eisler at UMB. And it was just, I mean, I will go more into it as I start my presentation, but it was just the very beginning of a new project she started working on just this summer. So it was very exciting to be able to be at the forefront and at the very beginning of this whole new project that I found super interesting. So my presentation, as you can see, is titled A New Testing Procedure for Detection of Borrelia burgdorferi. And for those of you who do not know, Borrelia burgdorferi is actually the bacteria that causes Lyme disease. Whoop. So just before I begin, I want to tell you guys a little bit about how my presentation will be broken down. I'll first introduce what my summer research was about, and then I'll be able to share a little bit more about how I was able to obtain this experience and how I actually got my position. Then I'll share how COVID was thrown into the mix, because obviously the story would not be complete without sharing how COVID changed everything for me this summer. And then I'll share a little bit about what my job as a research associate in the Eisler group entailed and what different experiences I obtained because of it. I'll share a little bit about what I was able to find my time there, and a little bit about what I thought about the overall experience. And I'll give a little bit of advice for all of those undergraduate students out there thinking about doing research. So this summer, I was able to work at UNB. I worked in FJ Tool Hall, which, as you guys already know, is the home of the chemistry department in our beloved university. My supervisor was Dr. Sarah Eisler, and the focus of my research was Borrelia burgdorferi's mechanism of infection. And we were hoping to find how we could make use of this mechanism to our benefit in order to be able to detect this bacteria. So the idea behind this research really came from what a big problem Lyme disease is around the world right now, but especially in North America, Europe, and Asia. And just to give you guys a better picture of how big this issue is, every year, the number of people becoming infected with Lyme disease and suffering because of it just keeps growing. One of the papers I was reading over the summer was talking about how in the United States alone, there was around 30,000 new cases every year being reported. And in Europe, there was around 85,000 new cases. And the research, or at least the data compilation that these people were doing, they were they intentionally were sharing about it, because it turns out that the numbers were an understatement of what the real numbers are like, because as you guys know, not everyone who gets sick actually gets compiled in the data, because maybe they don't know they're sick, or they don't actually, they don't get jotted down in the data per se. And the issue is that these numbers are expected to continue increasing as time goes on. So another factor that I think is very important for you guys to understand the why behind this research is that some of the current testing methods, or the methods that are used to test for Lyme disease or Borrelia burgdorferi in people, make it really challenging to give people an accurate result on whether or not they actually have Lyme disease. And I'll tell you guys a little more about why these methods make it so challenging throughout my presentation. But this whole experiment that we were doing over this summer was Dr. Eisler's way of starting a new project that could hopefully address some of these issues, and then also create at the same time something that had really never been done before. But before I go deeper into my research and what I did over the summer, I want to backtrack a little bit and tell you guys the story from the very start. So when I first came to Canada, I'm actually, just a little background information. I'm actually an international student. My home country is Honduras in Central America. So I decided to come to Canada to study science because I love research. And from the very start, research was something that I was thinking about and I was considering. And when I started, I already knew that research was something I would potentially end up doing, but it wasn't until my third year that I actually started asking myself more serious questions. And part of that came from the fact that my undergrad career was almost over. So then I wanted to start asking myself more serious questions, just like, do I really want to do research? Is it really for me? Is it something I would enjoy? Why would I want to do research? And if I do research, what type of branches of science would I pursue? So another thing about me is that I actually love to ask people questions, not just myself, especially if I know that the people have more experience than myself. So I remember in my third year, most of my lab TAs were master's students. So sometimes I would just randomly approach them and be like, so how did you decide that you wanted to do a master's degree? Or like, what is your research like? And every time I talked to them, I realized that I would come off like feeling more inspired because many of the things they would mention, I never would have thought about them on my own. And I was like, I can't believe like that is something you can actually do. Wow, that is so interesting. And then one time I remember I was sitting with my academic advisor and I was basically saying, okay, so I know I don't want to do med or dental school, and research is actually something that I am legitimately interested in, but I just still have so many questions regarding it. So he was super helpful because he helped me first narrow down what areas of research I might potentially be interested in. And then he just basically told me, well, if you don't try, then you'll never really know. You should probably just try working with one of your professors for one term and then see how that experience helps you figure out whether research really is something for you or isn't. And then he also told me the way that you would go about it is, like Jake was mentioning earlier, you have to apply for a grant and then you obtain funding for it. The issue was the only funding that I had heard about at the time was the NSERC USRA, like Jake was telling you guys earlier during this conference. And the issue is that some of the requirements, like he said, is first having a very high GPA, but also you have to be a Canadian permanent resident or citizen in order to apply. And obviously I wasn't, so that was then something I could not pursue. And I was basically just like, okay, well, there goes my chance at ever even realizing whether research is something for me while I'm an undergrad. But what was awesome was that I actually was sitting with my organic chemistry professor actually another day, and I was just asking about an assignment of all things, and we were just like trying to figure out whatever issue I was having with that assignment at the time. And once I was done asking whatever questions I needed to ask, I remember I was just like, you know what, whatever, like, Dr. Eisler, how did you realize you wanted to be a researcher? And so she shared a little bit about her story with me and her experience in her decisions when she was an undergrad. And then I was also able to share a little bit about my own story and like how I wanted to do research, and I couldn't really get funding because I wasn't a Canadian citizen. And then she was basically just like, well, you don't really have to apply to an NSERC. There are other funding opportunities out there. Shocking for me at the time, but anyways. And she told me, well, you could always apply to one of them. Is called the New Brunswick Health Research Foundation. And the only thing that you'll have to keep in mind is that your research will have to be somewhat health related. And that was fine by me because that is kind of the type of research that I was interested in anyway. So then as the term went on, I realized that I was really enjoying Dr. Eisler's class, and that, well, first of all, she already knew that I wanted to do research for the summer. So then I just approached her eventually and I asked her if I could join her research group for that summer and if we could apply for the MBHRF together. So we just went through that whole process, and eventually I got my grant and I was able to work with her group for the summer. So like I said before, my story really would not be complete if I didn't share how COVID affected my experience. And obviously COVID changed everything for everyone all over the world, but in particular, for the sake of this story, I will share that things were actually ended up being drastically different than what we were first anticipating. Because when I first joined the group and when I received my grant, Dr. Eisler and I started talking about what my project was going to be looking like. And the original purpose for my summer was to start synthesizing a fluorescent molecule that we could then use in the Lyme testing kit that she was planning on designing, so that the fluorescent molecule would be able to indicate the presence of Borrelia burgdorferi in whatever samples we were looking at. But we found some very key obstacles in order to continue with this experiment. And first of all, it turns out that my time in the lab was cut a little shorter than we needed because there were quite a few rules that needed to be set in place and parameters in order for researchers to be able to go back on campus. So I couldn't do all of my time in the lab, and I lost at least like a month where I wasn't going to the lab and I was just doing research at home, reading a bunch of papers about different strategies that we could use and different organic synthesis mechanisms that we could use to create our new fluorescent molecule. And then the other issue was that the starting material that we would need in order to actually synthesize the molecule was taking forever to arrive because, of course, COVID got in the way of all of the shipping and whatnot. So because of all of these obstacles, Dr. Eisler and I had to reassess what my summer was going to end up looking like. And our first hope was that we would actually be able to continue with our synthesis experiment. And that meant that I was able to continue going to the lab and practice some techniques that I would then need once my synthesis experiment started. But the second thing that we would do is we would start a side project where, because of my biology chemistry background, so because she is an organic synthesis chemist, and many of the students in her lab were mostly just doing chemistry stuff, I was the person that had the most biology background there. So my job would also be doing some research on the biology behind Lyme disease and how Borrelia burgdorferi infects new hosts. And so then with all this information, we would be able to design the way we would do our testing kit and how those methods would proceed. And this is a funny picture of myself in the lab. Remember, this was the middle of the summer, and I barely took any pictures of what I was doing because I'm not great at taking pictures, but there was just this one day and I was feeling inspired because, as you can see, the whole getup that we had to use while we were there during the summer was, obviously, if you've been in the lab before, you know that you have to use the lab coat and goggles. But on top of that, we had to wear masks. If I was doing training, so like if Dr. Eisler was actually teaching me to do stuff inside of my fume hood, I also had to wear this super uncomfortable face shield that would make me super sweaty and feel super gross. And anyways, I was just feeling inspired and I wanted to send a picture to my sister back home. So I literally just like snapped a selfie and I wrote the little caption, "Science in times of COVID." And she didn't appreciate it half as much as I did. But anyways, I thought maybe you guys would. So I just decided to give you guys a little taste of what my summer looked like when I was in the lab. And besides wearing an incredible fashion statement while I was in the lab, part of my job, besides, well, being a research associate, was doing weekly online meetings with the Eisler group, where different students would talk about their own experiments and how their progress was going. And there was quite a few of them actually working with Dr. Eisler. So there was a few master students, a few PhD students, and a few of us undergraduate students also working with her. So it was just an amazing opportunity to meet more people doing awesome research and just like-minded individuals doing, or well, loving science. So that was probably one of my favorite parts about the whole experience, just getting to know new people. And then also it meant that I would have weekly online meetings with Dr. Eisler where we'd discuss how my project was going and how my research was doing. And like I said before, I also went through some training on organic synthesis techniques, which was pretty cool because these were techniques that I hadn't learned before because I had taken organic chemistry labs in the past, but the techniques that we would be using in the lab went above and beyond anything I had done before, and that was pretty cool because otherwise I would never have experienced anything like it. And the most part of my summer was taken up by reading journal articles and finding out more about Lyme disease and Borrelia burgdorferi and using this information to be able to come up with a design for the testing kit. And I honestly learned so much through reading all those articles and just immersing myself in so many rabbit holes. But the thing I loved the most about it is that I was actually able to find information that would be super relevant for making a design eventually once this project actually was able to kick off. And before I start telling you guys more about my findings, I just want to give a little background information on Lyme disease to make sure everyone is on the same page. So, as I mentioned before, Lyme disease is caused by becoming infected with Borrelia burgdorferi. And this isn't just one species of bacteria. This is actually the full name of this group of bacteria is Borrelia burgdorferi sensu-lato, And then the species that you can find in North America is called Borrelia burgdorferi sensu stricto. And the species that you can find in Europe and Asia are a little more varied. They're Borrelia abstelli, Borrelia garini, Borrelia spalmani, and Borrelia bavarensis. But all of these different species infect a new host in the exact same mechanism, and that is through being introduced to a new body through the bite of a vector. And in this case, the vectors are the ticks of the species Ixodes pacificus and Ixodes capillaris. And these are more commonly known as the black-legged ticks. An interesting fact about these ticks, though, is that they have to be attached to somebody's, whatever organism's body for 36 to 48 hours in order for Borrelia burgdorferi to actually enter the person's bloodstream. And the reason I'm mentioning this is because this became super relevant later on when I was looking into other things for the testing kit. So some of the things that can occur when somebody does become infected with Borrelia burgdorferi actually can be broken down into 3 different stages. The first stage is called early localized stage, which is occurring between the first few days after becoming infected with this bacteria. And some of the key things that you can see is a skin rash, which is known as erythema migrans. And it is actually characterized because it looks like a bull's eye. And some people think that this is a very key thing to note and to be able to tell for sure if somebody is infected with Lyme disease. But the issue with it is that not everyone gets this little rash. And then the second, well, a few other things that can happen is you can get fevers, headaches, and muscle and joint pain. Then in the second stage, called the early disseminated stage, occurs during the first few weeks after becoming infected with the bacteria. And some of the things that can happen is you can get nerve palsies, meningitis, fatigue, dizziness, and fainting on top of the previous symptoms that will not disappear. And then the last thing is in the very last stage, which can take up to years in order to actually express itself, is that you can see the emergence of arthritis in large joints, and the central nervous system can actually also become compromised. And the sad thing about all of these different stages is the fact that sometimes they overlap, sometimes some stages are skipped, and in the worst case scenario, people don't show any symptoms until they reach the late disseminated stage. And that is actually a very, very tragic scenario because this is the hardest stage to treat people at in order to get rid of Borrelia burgdorferi in their bodies. So like I mentioned before, there are a few testing methods that are currently used to detect Borrelia burgdorferi in people's bodies, and there are some challenges that come with them. But first, I just want to mention the 2 official ones that are currently used around the world in hospitals and whatnot. And the first one is the standard 2-tire testing procedure, which uses an enzyme-linked immunosorbent assay, which is or also known as ELISA. And this testing procedure actually looks for antibodies in people's bodies. So then the second thing that they'll do immediately after an ELISA is a Western blot, which also looks at the antibodies that are produced and present in people's bloodstream or blood. And then the second official testing procedure that has come out recently in the past few years is the modified 2-tire testing. And this one is really just comprised of ELISAs. So as you can see, one of the main ones, or one of the main companies that has come up with different ELISAs is CU Scientific. And basically the first stage is you just do an ELISA, and then you can either follow that with a Western blot, or you can follow it with other ELISAs that are made from the same company. The issues with this method is, first of all, that you can get false positives. And this means that you can obtain a testing procedure that tells you you do have Lyme disease, but it turns out that you actually don't. And this is actually possible if a patient that is suspected of having Lyme disease is actually infected with other bacteria that will produce similar antibodies in their blood. And then you can also get false negatives, which means that you actually do have Lyme disease, but the test will tell you that you don't. And this happens when a patient is tested too early after being infected with the disease. And basically, the reason this happens is because your body will take a few weeks in order to produce the antibodies that this test looks for. So if you're tested too soon after being bitten by a tick, the antibodies will not show, and you'll be told that you don't have Lyme disease. So our idea for creating a new testing procedure really at the heart of it was just doing a test that could address the issues I was telling you guys about before. And some of the expectations that we had for this new procedure was, one, that it would be simple enough for the public to use. So if people wanted to test themselves, they would be able to do this. And the second thing was that there would be no need for blood extraction, and the desire for this expectation to come in was twofold. First of all, we didn't want to rely on antibodies in the blood, because that was the whole issue with the other testing procedures that are currently being used. And the second one was, if we want the public to actually be able to do this, we don't want them to go through the hassle of having to remove some of their own blood. And the third thing was, like I mentioned before, that we wanted to use a fluorescent molecule in order to detect Borrelia burgdorferi in whatever samples we would be testing. So then all of this meant that we had to find a new way to detect Borrelia burgdorferi that would not be linked to the person themselves, but actually to the ticks. We had to find a way to use the ticks to detect Borrelia burgdorferi. And the way we decided to go about it is we decided to look into the proteins that are expressed by Borrelia burgdorferi. And here in this slide, you can see I'm sharing one of the tables that I was working out throughout the summer. And I was basically just writing down a whole bunch of different proteins that are expressed by this bacteria and what they do. And this is just what I was able to narrow it down to as I was going on with my research. And eventually, I was actually able to narrow it down even more, because it turns out I found that 2 of these proteins are actually very intricately connected. And what happens is that the ticks, well, not the ticks themselves, but the bacteria actually needs to express OSPC in order to be able to infect a new host. And the issue with this is that OSPC is not always being expressed in their bodies. What is actually being expressed is OSPA. And the reason this is relevant is that when OSPC is being expressed, it is then that the bacteria will be able to move on to a new host. And the way this works is the tick will attach to a new host, and they will start eating blood, basically. And once the blood comes into their bodies, then that is when the temperature inside of them starts to change, as well as the pH. And this will be the signals that the bacteria will take onto itself to stop producing OSPA and then start producing OSPC instead. And like I mentioned before, the tick has to be attached a certain amount of time in order for the bacteria to move into the new host's body, because OSPC takes around 36 to 48 hours to reach its maximum production per se, and that is when the bacteria will actually move into the new host's body. And so I just added a few pictures of what OSPC and OSPA look like, but I don't want to describe them anymore because, as you can see, they're actually pretty complex. And the other thing that we wanted to talk, to find in my research was one molecule that we could make sure was simple enough that our fluorescent molecule could also bind to. And the issue was, when we started looking for proteins that we could use as our way to identify Borrelia burgdorferi, was that we also recognized that we would have to find a way to bind that protein to the fluorescent molecule that we were hoping to produce in the lab. And if you guys don't know a lot about protein binding molecules, it's actually a very complex process, and it's very, very specific. Proteins won't just bind anything. So that was a very big challenge that we were facing when we were starting off with this experiment. So what we decided to do instead was find a simpler molecule that the proteins were already known to bind to, and then hopefully the molecule would also be simple enough to also be able to interact with the molecule that we were hoping to produce. So then what I loved about OSPC, and actually I was scared that I wouldn't find anything when Dr. Eisler showed me those instructions, what was awesome about OSPC is that it actually exactly fit the bill, because OSPC, the way the bacteria infects the new host is that it uses OSPC to bind to the extracellular matrix. And specifically, it binds to this molecule that I added here called dermatan sulfate. And as you can see, it's relatively simple because as you can see, it's just like, it has quite a few OH groups. And as Dr. Eisler said, it should be simple enough to synthesize in the lab. And it also has quite a few groups that we can use to attach our fluorescent molecule to. So I was super excited to find both about OSPC and OSPA and their relationship, and also about dermatan sulfate. And by the time I found all of this information, my 4 months in the lab were also coming to an end. So although I was sad that my experience was ending and that it was so different than what we were originally hoping for, I was really happy that I was actually able to leave concrete information that whoever took on this project next could also use to continue with the experiment. And basically, my biggest 2 findings would have been the relationship between OSBA and OSPC, and how this amazing relationship would not just tell us of the presence of Borrelia burgdorferi, but it would also tell us not just that it's there, but also that it's actually infectious, because we want to be able to tell if people actually got infected with the disease and not just the fact that the protein in the bacteria itself is there. And the other thing that I found, like I mentioned before, is just dermatan sulfate, which will make the job of synthesizing our molecule and just the interaction between the protein, dermatan sulfate, and our fluorescent molecule all that much simpler to deal with. So I just want to share a little bit about what I thought about my overall experience. And quite frankly, I would have no complaints if it weren't for COVID. COVID just changed a lot of things. But even with that, I was able to fulfill my dream of being part of a real research lab in the real science world. And that was pretty amazing for me, because it was something that I had been hoping for probably since I was like 5 years old. And then the second thing that I loved about this experience was that I was actually able to narrow down what ideas of the type of research I want to pursue would look like. And lastly, I was able to connect with some amazing people that just love science and are super kind and super willing to help, and just make some great connections. And like I said before, this was very rewarding for me because of all the findings that I was able to obtain and all the information that I was able to leave behind for whoever takes on this project next with Dr. Eisler to continue. I would also like to finish my presentation with some advice for undergraduates that maybe find themselves in a similar position as I was in a few months ago, well, around a year ago. And the first thing I would say is, don't be afraid to start or keep asking questions, because that was a big thing for me. That is what actually carved the path that I ended up following for my 2020 summer. And it can seem kind of silly, because a lot of times it just seems like there's this idea that as science undergrads, we kind of need to have things figured out real quick. But what I learned is that there's just so many different opportunities that you can take, and so many different paths, that having everything figured out in your undergrad would actually limit what you would be able to pursue in the future. The next thing I would like to advise you guys with is, don't be afraid to connect with your professors, because these are the people that are actually best able to give you advice about what research is like, and would actually be able to help you get opportunities like the one I had with my organic chemistry professor. And the next thing I would say is, don't count yourself out, because that was one of the things that I was about to do last year, or even a few years previous, actually, when I heard about NSERC and not be able to apply for it, even though I had a great GPA. So again, just don't count yourself out, keep asking questions. There are so many more opportunities than you even realize, and they're all equally great. And the last thing I would say is, research actually doesn't always turn out the way you expect. The number of times I've heard researchers tell me that research, well, at least half of the research experience is troubleshooting. And there's so many things that can go wrong. So I would say, just don't be afraid with research going wonky, because that is part of the experience, and that is part of what makes it so great, just being able to find new science, and also just being able to solve problems with such ease. So whatever happens, make the most of it anyway. In my case, most of my problems actually came from COVID, but even with that, I was able to do some science that I was really proud of and that I really actually enjoyed. So here's a list of my references that I will not bore you with, and I'll be happy to answer any questions that you guys might have.
All right, so if anybody has any questions, just raise your hand.
I have a question for you, Melina.
Awesome.
So you mentioned that, I was like a little bit confused. So you said the protein after you get a tick bite enters like 36 hours, 24 to 36 hours after you get it?
Yeah, so like what I was trying to say is the bacteria will not express the protein. Well, it will start expressing it, but it will not reach like its peak up until like 36 hours after the tick starts feeding. And so you need a certain amount of OSPC in order to actually enter the host. So that is what I meant with what I said. The protein,
So if
it's in the genes, but it's not yet being expressed.
so if you realize that you had a tick bite like an hour after you got it, and you went and got like the tick removed, would it still be expressed 36 hours later?
You probably would not see the protein there. There actually the research that I was reading on that, they were testing the presence of OSPC certain amount of time after the tick was fed. And even after 12 hours of the tick being attached to mouse was what they were using, the presence of OSPC was almost negligible. So if you tested it just 1 hour afterwards, you wouldn't probably find it.
And would you still get Lyme disease then? Or?
No, you wouldn't get Lyme disease because the bacteria would not actually be able to enter your blood because it doesn't have OSPC yet expressed in enough.
Okay.
Mm-hmm.
That's really neat. And great presentation. Thanks a lot.
No worries.
There's a question from an anonymous person. Would you happen to know of any profs that you would recommend doing health-related research if you're a biology student? I'm assuming at UNB.
Yeah, so actually, I don't know a whole lot. I actually never thought I would even do health research with Dr. Eisler because, like I said, she was an organic chemistry prof. But I think really honestly, I would just recommend asking around. I feel like you would probably find even more opportunities in the biology department. I do know that there's another professor in chemistry. Her name is Dr. Anna Ignasak, and she also is working on another mechanism to detect Lyme disease. So honestly, there are a lot of opportunities. You'll just need to find them. But I'm sorry, I can't give you more information on that.
Jackson said there's a lot of awesome health research going on at Dal Med NB as well, just if anybody's wondering. I had a question. Are you continuing with this prof, Dr. Eisler, or are you going to move on to someone else in the future to work on different research?
Yeah, so my hopes was actually that I would be able to continue with this research just as my side job during this term. And we still haven't figured things out with Dr. Eisler. That is still a hope. But right now I'm working on my thesis with Dr. Yanku. So that takes a lot of my time right now. But yeah, hopefully I also will be able to work on this too.
Cool. We have time for a 5-minute break if nobody else has any more questions before the next panelist. Thank you, Melina.
No worries. Thank you.
Melina, I don't know if you're still there because there's another question for you.
Oh, no. Yeah, I can answer that.
Somebody asked, would you be able to tell a little more about international students and the med school thing, please?
I think international students in med schools pretty much work the same way as for like Canadian students. Like you basically have to go through the same process. I know like international student fees in themselves, just as an undergrad, are normally higher than for Canadian residents, but I don't know how that would work for med school. Most universities will actually have all that information in the webpage, so I would just recommend for them to go look into that wherever they want to go. But yeah.
Awesome. So I think it's time to introduce our fourth and final speaker. Dr. Daryl Haggard is an associate professor of physics at McGill University, and she's a member of the McGill Space Institute. Haggard was awarded the 2020 Breakthrough Prize in Fundamental Physics as a collaborator of the Event Horizon Telescope for the first image of the hot plasma and photons at the edge of a black hole. She holds a Canada Research Chair, Tier 2 in Multi-Messenger Astrophysics and was a SCIspire Azraeli Global Scholar in the Gravity and Extreme Universe program. She completed a PhD and Master of Science in Astronomy at the University of Washington. So welcome, Dr. Daryl Haggard.
Hi, everybody. Can you hear me okay?
Yeah.
Excellent. It's a pleasure to be here with everyone today. Thanks for having me. I'm going to go ahead and share my screen if all goes well. Can we all see a beautiful black hole?
Yep. See a black hole.
Okay, great.
Yeah,
Great, glad to hear it. Okay, so I'm going to be talking about black holes, my favorite subject in the universe. And I just wanted to say thank you very much. It's a really nice to be with everybody today. So I'm going to talk a little bit about how we observe black holes, which sounds a little bit like a contradiction in terms. But we do actually see the hot plasma around black holes, and that's the kind of thing you're seeing when you look at this beautiful black hole in the galaxy M87. So I'm going to start. I think that you all asked me to just say a little bit about myself, so I'll just start telling you a little bit about my own background, and then I'll dive in and talk about the really cool research. So I am originally from New Mexico. So you're looking here at a New Mexican flag on this slide. I did my undergraduate degree actually in philosophy with a minor in mathematics. And so I had sort of a classics degree, which is my origin story. And I retrained into physics in a master's program at San Francisco State University. And then I went on and I did another master's and a PhD in astronomy at the University of Washington in Seattle, Washington. And then I moved from there into a series of different academic positions. So I don't know whether you all have talked to other postdocs today at all, but the path, at least for physicists, is typically after you complete your PhD, you do a postdoctoral fellowship or a postdoctoral position for somewhere between 2 and 5 years before you move into the professoriate, if that's what your career path and career desires look like. So I took my first postdoctoral fellowship at Northwestern University just outside of Chicago. And then I spent 1 year as a professor on the faculty at Amherst College in Massachusetts. And I've been at McGill for the last 5 and a little bit years. I spend quite a lot of my time doing outreach and mentoring of student researchers. So this is really something I take pleasure in and have spent a lot of my time doing. I also, in the years in between studying philosophy and going back to study physics, I spent time working in Silicon Valley and living abroad in China. And so that was a formative time for me as well. I encourage everyone to try lots of things out. Don't feel like you have to be too tracked in your career. It's good to go and explore and see the world a little bit. I also work quite a bit on equity, diversity, and inclusion in STEM. This is something I've been engaged with for many, many years. It's certainly been an important topic of conversation for everyone around the Black Lives Matter movement and here indigeneity and understanding how to be more inclusive as a field. So I care a lot about that. I'm a member of the Equity, Inclusion, and Diversity Committee for the Canadian Astronomical Society. I've worked previously for the Committee on the Status of Women in Astronomy for the American Astronomical Society, and also tried to think a little bit about governance and how we can do better to be inclusive as a professional society with the American Astronomical Society. I've also been a part of the Women in Science and EDI committees in the McGill Physics Department, and I spend quite a bit of time on my own self-study. So many of the things, I come from a majority white population, and that means that even though I am a woman in science, I just don't have the shared experience of a lot of people of color in the sciences. And so I spend quite a bit of time trying to understand what the literature says, what the personal accounts say, how I can do better, be a better mentor, be a better faculty member, be a better community member. My group publishes a ton of science. We have 65 refereed papers and thousands of citations. These are metrics that maybe some of you are familiar with, maybe you're not. But we also spend a lot of our time writing observing proposals. This is sort of a specific thing to my niche of astrophysics. As I mentioned, I'm an observational astrophysicist, so I need access to these observatories that are located all over the globe and often in space in order to do the kind of science that I love. And a lot of this requires resources in terms of people and funding, etc. So this was just a little slide to say that I have this kind of wonky background where I studied, Plato's laws of, physics, and I read the Principia and all sorts of funny other things that normally aren't maybe the path to a physics degree, but I really loved and respected and really took in this first education that I had in classics. And I should also really give a shout out to the amazing people I work with. So this is a little look at my research group at McGill. So I work with two other faculty members in the Montreal area, Professor Tracy Webb and Professor Julie Labachik-Larando. Tracy is Professor Webb is a faculty member with me at McGill, and Professor Labachik-Larando is a professor at the University of Montreal. And so the three of us do quite a bit of work on black holes and how black holes and galaxies co-evolve across cosmic time. Here's my current postdoctoral fellow who just came across the border yesterday. I'm so excited. The border closure between the US and Canada has been, I think, correctly in place, but made it very difficult for international students and international postdocs to come and work across our two nations. These are my wonderful graduate students down here: Hope Boyce, Sud Al-Karouzi, Constanza Echiburu, Teofil Begin, Nicolas Vieira, Anna Liu, and Katie Savara is actually an undergraduate working my group, but she's got a photo on here because she spent so much time over the last year and a half working with my team. I also have some recent members here, other wonderful scientists who've been a part of my group, and I currently have about 8 undergrads working on the team. And so this is really it's a big, robust, fun group. We're all online and remote the way all of you are. That has its challenges, but we do a lot of Slack channeling and a lot of Zooming. And so far, it's maintained, I think, all of our sanity to have each other to support and to think about science and do cool science. All right, so that's the introductory stuff. I'm happy to chat more about career path and career trajectories and team members, et cetera, toward the end. But let me spend at least the next 20 or 30 minutes telling you about the science, at least some of the exciting science that I've worked on with these wonderful group of people over the last few years. So this is this amazing black hole. We call it M87 star. We give these things stars when they are the brightest radio source. I'll show you some radio images in a second, in a galaxy. So in our own Milky Way, we have a supermassive black hole. It's in the direction of the constellation Sagittarius, and we call it Sagittarius A star. The capital A is because it's the brightest radio source, and the star is because it's in the center of the galaxy. So this black hole that you're looking at here is in a nearby galaxy. The galaxy is called M87. And so the black hole is called M87 star. So this is technically M87 star, but by far the more interesting name for this is the one that the native indigenous Hawaiian peoples gave this amazing black hole. Many of these images were collected, or at least part of the network that collected these images as a part of the Event Horizon Telescope is on the sacred mountain Mauna Kea. And so this black hole is called Poehi, which is from one of the creation poems of the native Hawaiian peoples. And it means the generative darkness of creation. It's a piece of one of their beautiful poems, which is the Kumulipo, which I may or may not be pronouncing correctly. So anyway, Poehi is the far more beautiful name for this incredible, incredible first image of a supermassive black hole. Okay, so let's talk a little bit about the black hole. So this black hole, this amazing supermassive black hole, has 6.5 billion times the mass of our own sun. So it is a real monster out there in the universe, lots of mass. But the thing that makes it a black hole is that this mass is packed down into a structure that is extraordinarily small compared to the scale of that mass. So this is a wonderful XKCD comic. You all have seen probably many of these, but I love it because it really shows off just how similar the scale of the solar system is to the scale of this incredibly massive black hole. So here is our sun. This is the orbit of Pluto, and this is the current position in XKCD comet format of Voyager 1, which is the most distant satellite we've ever launched off the surface of our Earth and sent into space. So you can see that if we happen to live at the center of this black hole, that we would barely have just now sort of explored the sort of extent of the black hole or the accretion flow of the black hole with our own little solar system scale. So I show this to you partly because it just shows how incredibly hard it is to explore the universe with actual physical objects like spaceships and satellites. But I show it to you also to impress upon you that what this means, since the black hole is down inside this hot plasma, that means that this 6.5 billion times the mass of our sun is packed into something that's about the same size as our solar system. So that's a lot of mass in a relatively tiny, tiny space in a universal kind of sense. So let's put that on a slightly more sort of tangible scale. So the Earth here is something like 3 millionths the mass of our sun. So this supermassive black hole is 6.5 billion times the mass of our sun. Our Earth is 3 millionths of the mass of our sun. But we could still make our Earth into a black hole if we could figure out how to smush all of the mass of the Earth. This includes your mom, your dad, your grandparents, uncles, aunts, all the trees, all the flowers, all the mantle, all the core. If we could smush that whole thing down into something that's roughly the size of a sugar cube or a rolling die, that would actually create the conditions that would make our Earth into a black hole. And so what this means is that black holes not only are, impressive and interesting objects, but they basically represent the most compact, dense objects that we can study in astrophysics. And so why is it a black hole? Well, the reason that we would consider an object like our Earth squished down to the scale of a sugar cube is that once you squish it down to be that small, the escape velocity, the velocity that something has to have in order to escape off the surface of the object. Say, for example, you want to throw a baseball. In order for your baseball to get off the surface of the Earth, it has to get up to something like 11 kilometers per second in order to escape the gravity of the Earth. But if you were to take the Earth and you were to squish all of that mass down into a much smaller size, you would create much higher surface gravity. And eventually you would reach the point where even something moving at the speed of light could not escape the surface of the compact object. So that's one of the definitions for a black hole, is that you've got an object that is so compact, so dense, has so much mass in such a small amount of space that even light, even photons cannot escape out of the gravitational pull of that object. So anyway, that's a kind of a fun, intuitive way to think about black holes. This is a beautiful video that was created right around the discovery of that supermassive black hole, or the image of the supermassive black hole. So we're going toward the constellation Virgo. One of the coolest things about this video is it's made entirely with real data, which is just phenomenal. So we're zooming in toward a galaxy cluster, which is the Virgo cluster of galaxies. In the center of Virgo is this really bright galaxy known as M87, and you can see that galaxy come into relief now. You can see it's got a bright jet coming out of the center, and right down at the origin of that incredibly impressive jet is this really, really, really compact object that's creating this very energetic outflow, this supermassive black hole. So this is a beautiful demonstration of how many generations of astronomers have worked together to accomplish this really, truly phenomenally sensitive imaging at extraordinarily small size scales. So one of the analogies people love to throw out there is that in order to take this picture, you have to be able to get the fineness of imaging that is equivalent to taking a snapshot of an orange sitting at the surface of the moon. So you have to really have incredible precision and very high sensitivity to be able to look at this radiation with this much detail at such a large distance. Okay, so we've already looked at that. So what's actually going on in this, in these images? Why does it look like that sort of sphere? I'm going to show you another movie in a second, but let me tell you what you're going to see first. So why does it look like a ring? It turns out there's a lot of mass in there. I already told you light can't escape the black hole itself. So the light that's really within the event horizon is trapped. So we're not seeing that light. That light is blocked from our view. It can't get out. But just outside of the black hole, there's more mass and dust and other stuff. And as it falls into this very deep potential well, it heats up to very, very, very hot temperatures, very high temperatures. And that's what we're seeing when we look at the light from M87 star or from other, we call them accreting supermassive black holes. But the light actually that's in the vicinity of the black hole, just outside the event horizon, is actually being bent by the very, very strong gravity of the black hole. And so what you're seeing here is an artist's impression. So this is not real data anymore. This is entirely simulated, this little video I'm going to show you. So you're seeing an artist's impression of the hot gas and dust, a little bit of that jet maybe being driven by the deep gravitational potential well of the black hole. And then we're going to actually dive all the way down toward the event horizon. We're going to see photons actually being bent by the incredibly strong gravity in the vicinity of the black hole. And that's what makes up the ring. So again, this is an artist's impression, a little sort of artist video of what might be going on. We're diving down through the accretion disk down toward where we can see the event horizon. The black hole's in here, the event horizon is here, and you can see this event horizon actually bends the trajectories of photons. It bends the trajectories of the light as it goes around. And right now we're looking at it from the side, and it's going to bend toward the perspective of the observer. And you can see that from our perspective, all of these photons, I guess from your perspective, they're all being bent around the black hole and kind of focused into this ring. And so this is a sort of simulation demonstration of how we end up having this sort of ring structure that we see when we look at a black hole, even a real black hole out in space. Okay, so this is a really super impressive object. It's very exciting. We are working very hard as a collaboration. So I should say this was given Science Breakthrough of the Year in 2019 when we first presented the image and made the discovery. But what I was about to say is that this is actually an incredible international effort. It takes, in order to make these really amazing high precision images, it takes telescopes spread across the entire surface of the Earth. We use the whole Earth as if it's our single telescope, where we only have little patches where we can actually see light. Those patches are where the telescopes are. So I mentioned that there's a variety of different telescopes around the world, including the CSO, which is near Caltech in California, the JCMT and SMA, which are both on top of Mauna Kea in Hawaii, as I noted. Karma is in California, and so is SMT. You can't see all of these things. IRAM is over in Europe, and APEX and ALMA are down in Chile. So these are telescopes, and actually we should have the LMT. Maybe this is the LMT. The LMT might not be on this picture, which is in Mexico. Oh no, this is the LMT, right smack in the middle. So these are amazing telescopes that actually network together using electronics and incredible computing capacity to operate as one single observatory to look into the sky. And so this collaboration is made up of people from around the entire world. So there are more than 200 scientists in the Event Horizon Telescope collaboration. We represent 60 institutes, 18 countries, 6 continents. My team just recently joined formally to this collaboration, and it's very exciting to join these incredibly bright people from everywhere. So it takes a tribe, and this tribe is really out there really trying to see not only M87 star, but another supermassive black hole. And that's the one here in the heart of our own Milky Way galaxy. So I'm hoping in the next sort of year and a half, we might be able to share images of our own supermassive black hole here in the Milky Way. It's a slightly smaller black hole. Instead of 6.5 billion times the mass of the sun, it has something like 4 million times the mass of our sun. That supermassive black hole, oh, I should have put a slide in here about this, but that supermassive black hole is called Sagittarius A star. And we know its mass to beautiful precision because we watch these stars orbit around it. And that discovery, the monitoring of those stars and the incredibly precise mass for our own supermassive black hole and tests of general relativity in its vicinity earned Reinhard Genzel and Andrea Getz the Nobel Prize in Physics just last week. So that's very exciting. Andrea Getz is a friend and a colleague of mine, and so I am just absolutely thrilled to have her in the position of a Nobel laureate. And so keep an eye on that space if you're really interested in supermassive black holes. Okay, so I mentioned that we see this jet in the center of M87, and I mentioned a few times that M87 star is observed in radio wavelengths. And so this is an amazing image going from the very smallest scales where we've taken this Event Horizon Telescope image of the supermassive black hole, and then zooming out a little. So the supermassive black hole is buried now down deep inside this bright emission. So this is some of that hot gas. We're looking at real data here. This is some of that hot gas, and you can start to see a kind of a collimated structure attached to it. If you zoom out even a little bit more, you can see now that this collimated structure really is quite narrow and very bright at radio wavelengths. And then if you zoom way out, you can see we've got an optical image in the background, and here's a radio image. Turns out this little tiny supermassive black hole is pushing out an incredible amount of energetic feedback emission out into the largest structures here that make these enormous bubbles. There's another jet going this direction, which we can't see as well because it's moving away from us instead of toward us. This one we can see it's moving toward us. And this incredible structure, this is way out beyond the edges of the galaxy. So this is the galaxy in here, and you can see this jet in its center. And this is how black holes and galaxies interact. We call it black hole feedback. And this interaction is really fascinating and not very well understood. We're still trying to understand how the two interact with one another and co-evolve in time. So this is extraordinary to be able to piece together all these different size scales and see what the whole environment looks like all the way from the sort of microcosm out to the real macrocosm of the galaxy and beyond. So these are radio wavelengths. My team often looks at the same structures at X-ray wavelengths. So this is now observing the same galaxy with the Chandra X-ray Observatory. You see slightly different things. Now you really are seeing hot gas encompassing the entire region around the supermassive black hole. Really interesting tendrils out here. These are magnetic structures. This jet itself is also a magnetic structure. Zooming in here a little bit more, you can see that the jet even has substructure within it. We think these are little tight knots of plasma that's been ejected out of the black hole and is collimated along this magnetic jet structure. So my team is what we call multi-wavelength and sometimes multi-messenger astronomers or astrophysicists. So we really are interested in understanding all these different wavelengths of light that we can use, the submillimeter where the Event Horizon Telescope is operating, radio wavelengths where many of the other observatories that were linked together to make the EHT also observe, and then space-based observatories that allow us to look at the highest energies and see the X-ray emission from these same structures. And of course, just to reemphasize, buried down here at this little cross is this amazing supermassive black hole that the Event Horizon Telescope has imaged. So of course, people would love to do better than just that little video that I showed you. This is a graphic showing you some of what might be going on as we try to link together these different images that we make and also the simulations we make in computers. So this is sort of our picture, our mind's eye picture of what we think is happening. There's a supermassive black hole in the middle, this hot accretion disk. This is hot material, gas, dust, etc., flowing in a sort of disk shape. Anything with angular momentum in space, as it spins, it tends to flatten. So you get sort of disc-like structures. And we believe that this is then connected down at the base, the inner regions of the disk and the spin of the black hole itself twist up magnetic fields and create these amazing jetted structures that I was showing you just a minute ago. And so down here at the horizon scales, we're seeing this Event Horizon Telescope image. And we think if we can sharpen this image over time, that we may be able to actually start to look at these sort of finer structures. These rings, these perfect rings here are called photon rings. These are the last orbits that photons can make before they end up getting trapped inside the supermassive black hole. And if they sit just outside of that event horizon, photons can orbit stably for very, very long periods of time and build up their emission, the light they give off. And so we see these amazing, really tidy looking photon rings. Some of the photons actually can come even closer to the event horizon. This is due to the rotation of the black hole in a general relativistic phenomena called frame dragging. And we'd like to figure out how to link all of this up to some of the observations that we make out at those larger scales, and understand how the black hole, the accretion disk, and the jet are all interconnected down at these smallest scales. So this is one of the objectives of the Event Horizon Telescope, and we're working very hard on all of the data that we've got in hand to try to understand it better. So this is just another image of the same. I think I'll skip over it, but just to say that, you know, we've worked very hard to try to understand the magnetic fields and the accretion disk and the jet and the black hole image to try to piece together all the different parts of the puzzle to understand really what's happening in this very exotic, very high gravity environment. And I wanted to just bring this back for one moment to the place that I started here talking about my background in philosophy and mathematics, just to say that this beautiful image is not a black hole and not an accretion disk. This is in fact a beautiful poster that was made from, I don't remember, it might be NASA that made this poster. It could be the European Space Agency or the European ground-based observatories. But this is the planet Earth here, and these are the orbits of all of the different spacecraft we've sent out to explore objects in our solar system. And this is what got me excited about science when I was an undergraduate. So I couldn't believe that you could take individual beautiful spacecraft and actually use the planets in our solar system to give them what we call a gravitational assist, to basically slingshot them out further and further into the solar system. And so you can see these gravitational assists. I'll just follow one trajectory here. Here's a satellite launched from the surface of our Earth. It actually goes around Venus to pick up a little bit of a extra gravitational boost. You can see here Venus gravity assist. And then it comes around and it gets a gravity assist by our own Earth. And then it gets flung out. In this case, it was one of the Cassini mission or Galileo. I don't remember which one I was tracing with my fingers, but these are missions to go out and explore the moons that are orbiting around Jupiter and Saturn. So this is what got me excited about studying physics and about studying science. And so these are orbits and gravity assists happening here in our solar system with physical bodies we can touch with our hands, but it's actually not dissimilar from these amazing sort of pictures we're drawing of photons that are, responding to the gravitational field of supermassive black holes, which is where my research has taken me today. So I'll talk just very briefly toward the end here about the other domain that my research group is really excited about thinking about and working in. Still black holes, still neutron stars, but in this case, we're talking about a different class of black holes. These are the little small ones that result from the explosions of massive stars. So these are, we call them stellar mass black holes instead of supermassive black holes. So stellar mass black holes have sort of tens, sometimes as many as hundreds of times the mass of our sun, where supermassive black holes have millions to billions of times the mass of our sun. And neutron stars also are sort of in that low mass regime. So neutron stars and black holes result from the sort of explosions of massive stars, and these are the remnants they leave behind. So this is an amazing discovery that was made a few years ago in 2017. What you're seeing here is a fade from an optical image from the Hubble Space Telescope to the X-ray image from the Chandra X-ray Observatory. You're seeing a beautiful galaxy here in the middle. You can see dust lanes and all of the galaxy structure when you look in the optical. And then when you fade to the X-ray, you can see that there's some bright emission at high energies from the black hole in the middle of this galaxy and a few other interesting sources. But the one that's got a box around it is kind of off to the side. And so this is an amazing discovery. It was our very, very first multi-messenger astrophysics discovery. And so this source, it turns out, is the merger of two neutron stars. And these two neutron stars, as they in-spiraled and smashed together in space, emitted this kind of radiation called gravitational radiation. And because when they smashed together, they also initiated an explosion that we call a kilonova, we observed it, like you're seeing here, also with electromagnetic radiation, which is how astronomers like me normally operate. So we call it multi-messenger because it's using both this new facility we have to detect gravitational radiation combined with our sort of more traditional astronomical technique of using electromagnetic radiation. So once again, this was another science breakthrough in 2017. This is this amazing explosion that resulted in the gravitational radiation being emitted, all sorts of electromagnetic radiation being emitted. This is the original explosion. And then a huge jet being driven also from this source that smashed into the medium in that galaxy nearby and created a very long-lived and very, very spectacular set of emission that we could observe over multiple years. So this multi-messenger astrophysics is very exciting. It's really being driven by these gravitational wave observatories, which are called LIGO and Virgo. LIGO is the Laser Interferometric Gravitational Wave Observatory. It's located at 2 sites in Washington in the United States, the state of Washington and the state of Louisiana. And then it has an Italian counterpart called Virgo. And those 3 interferometers, they operate like Michelson interferometers. If any of you have taken a physics class where you discussed interference and laser interference. So this is basically exactly like a Michelson interferometer, but just a really, really big. The 2 arms of these interferometers are kilometers long, and they can actually detect gravitational radiation causes our Earth to get slightly squeezed or extended as the radiation passes over our, it slightly squeezes and compresses space-time. And so as the wave comes across the Earth responds to that change in space-time. And the changes it can detect are a fraction of the width of an atom. Really just incredible technology, beautiful optics, really phenomenal detectors. And they've detected a whole bunch of black holes merging, which we have never seen to have an electromagnetic counterpart, and a few neutron stars merging. So you can see here down here are these 2 different detections they've made of neutron star mergers, which we think, have these electromagnetic counterparts. So my team worked hard on that first one that I showed you the pretty image, the fate of. So this was 2 neutron stars that merged together. We don't know whether the remnant that they left behind was a black hole or a neutron star itself. We think probably a black hole because it was massive enough, but jury is still out on exactly what it was. And then my team also worked recently on another kind of sort of newbie merger where you have a neutron star and a black hole. We're not completely sure that one of the objects was a neutron star. It might have been a black hole that was just very, very small, or maybe it was a neutron star. And we thought, oh, well, if you have a neutron star in orbit around a black hole and the 2 of them crash together, maybe when the neutron star is sort of shredded apart as it's falling into the black hole, perhaps that would be electromagnetically bright, and we might be able to make a second multi-messenger detection. So my team spent a bunch of time following up. So we went after this source here. We tried to find it on the sky and then use the Canada, France, Hawaii telescope to tile over a very large area of the sky to see if we could see any flashes, any bumps in the night. And so this is work done by my fantastic grad student, Nick Vieira, and a postdoc working with me at the time, John Runn. And so these little colored patches are where we pointed the observatory. You can see they're really intense mosaics of lots and lots of pointings of the observatory to try to see what's happening. And these kind of contours and colors are showing you the highest likelihood position on the sky for where that gravitational wave source was. So we were not successful. We actually did not detect an electromagnetic counterpart in this case. We searched over 100 candidates and used all sorts of cool machine learning techniques to try to piece out anything new in the data. But there was no counterpart detected in this particular case. So this is sort of the really exciting payoff, and then also just the hard work of doing the science of trying to find these new sorts of black holes out in the universe. So this is ongoing work. People are still really interested in following up these sources. The LIGO Virgo observatories are closed down right now due to engineering upgrades and also in part due to coronavirus. Many of our ground-based observatories have been shuttered for many months, like a lot of the other operations all over the world. So we're hoping to get things back up and running after the engineering runs in about a year's time, and to be able to start doing this exciting science again. So I'm going to leave it there. This is my t-shirt that my team members and I all bought and wear together this year in order to kind of keep ourselves together. And I also want to just give one more shout out back to the very beginning of my talk as well, and just say, we're all part of society here. So black holes are awesome, and Black Lives Matter and Indigenous lives matter. And please be a part of those social movements as you study your science and do more cool things in the universe. So with that, I'll stop and take questions.
Wow, that was amazing. Does anybody have any questions? Jackson has a question.
That was a really great presentation. Thank you. I kind of wanted to extend off of what you said in your last slide there, and talk a little bit about diversity, equity, and inclusion in STEM.
Sure.
So I often find myself recognizing the importance of these ideas, but also realizing that there's so much I don't understand, and so much more that I feel like I need to learn. So what advice would you have for an undergraduate beginning to enter the world of research that wants to learn more about these ideas and promote them, but doesn't really know how?
Yeah, it's a super good question. So I think we all struggle with this, and I don't know what kind of a background you have, but particularly if you happen to be a majority population person, it can feel very uncertain. So listen a lot and look around and see where you can help and where you can plug in. So our community, and this is a community of amazing students. It's a combination of undergraduates and graduates and postdoctoral fellows and faculty. We're watching all of the protests over the murder of George Floyd, and we have, recent tragedy here with this amazing Indigenous woman, Joyce, in Quebec. And, we sit and we watch these things and we just feel powerless in the face of them. But it turns out that if you talk to your community and find out, well, so what are the barriers? Why are people not in my community? Why is my community not doing better? And talk to your colleagues and friends. And in our department, we've had an incredible initiative to create a sort of departmental policy to think about being more inclusive, recruiting better, talking to our Indigenous communities more, and finding out whether or not there are students who would be excited to join us in any one of our STEM disciplines. It doesn't have to be physics. And so there are a lot of initiatives. So we also do a ton of outreach, which I think can be a great way of just sort of dipping your toe in and starting to learn about this. So if you have an outreach component in the institution where you are now, public lectures, learning how to do science communication, it can make a huge difference for learning how to talk to people, especially around sticky issues. Read a lot. We have an amazing resource list that if you haven't found one already, I'd be happy to share. Read, self-educate, talk to people, and try to listen for where you can plug in and help, and just try not to be afraid. You'll do things wrong. I do things wrong all the time. And you just have to apologize and say, I did it wrong. I didn't know how best to be your ally. I'm sorry. And take a deep breath and keep at it, because I think that we have to get more robust to make this better, and we have to get more brave. So be brave, dive in, just try it all out. You'll just feel better for having, kind of ventured into a new domain and to learn something new. So I think there's lots of concrete things you can do. Read stuff, start a reading group, talk to your peers, talk to your community, do outreach, talk to your institution, find out what they're doing. There's lots around you that you can join.
That's awesome. Thank you.
There's another question from the Q&A. She loved your presentation, or they loved your presentation. She asked, is it easier to take photos of black holes from faraway galaxies than ones within the Milky Way, or is there another reason why M87 was photographed before the one in the Milky Way?
That's a super good question. So yes and yes. There are two reasons. Yes, it's harder to take pictures of things in our Milky Way. So my favorite analogy is a pizza. So imagine that you're you're a mushroom on your veggie pizza, and you want to look toward the middle of the pizza, but you've got to look through all of the cheese and all of the crap and all of the dough and everything else. So that's us in our Milky Way. Our Milky Way is a spiral galaxy. It's sort of a disk. It's flattened like many other things that rotate. And we live in that disk, out kind of in the suburbs. And we have to look through all of the junk of the galaxy in order to look toward our supermassive black hole in the center. And so we call this absorption or extinction. You have to look through gas and dust and stars. There's so many stars, which is amazing, but also very confusing. We literally call it confusion. So it's hard to find a supermassive black hole, which is super tiny on the sky, and to really be confident that you're seeing emission onto that black hole and not some other phenomena either between you and the black hole, or just that's complicated in the messy environment near the black hole. So looking through our Milky Way galaxy is hugely challenging, very, very challenging, and one of the major stumbling blocks to giving you a beautiful picture of our black hole. So that's a fabulous insight on your part. Absolutely true. The second thing, remember I said M87 star is 6.5 billion times the mass of our sun. Sagittarius A star, our Milky Way's black hole, is 4.3 million times the mass of the sun. So And black hole sizes roughly scale with their mass. So our black hole is smaller, but it's also much closer. And those two things are offsetting. So we actually think that Sagittarius A star and M87 star are about the same angular size. They look in projection like they're about the same size on the sky to us, because one is smaller, but it's also closer. But the tricky thing about it being smaller is that the stuff that's orbiting around it is orbiting much more quickly, because it's kind of closer into this black hole, and its variability timescales are very short. So over the course of one observing night, that beautiful static image you've seen of M87 for Sagittarius A star, it changes over the course of the night. And so that's really awesome, because maybe we'll be able to make a movie, but it makes it really hard for us to be confident that we've actually taken an image and it's not just some blobby thing that's moving around in the night. So the variability for Sagittarius A star and the fact that we have to look through the Milky Way galaxy are two of the big challenges. So as I said, I just recently joined the Event Horizon Telescope, and we are grappling, I mean, the collaboration formally, and we are grappling with that second question right now. So super good question.
Are there any other questions?
Yeah, Dr. Haggard. I was wondering, so as part of the Event Horizon Telescope Network, do you also work with space telescopes? Because I know you had mentioned a lot of like land-based telescopes, but would you be working with like the Hubble or any like satellite-based telescopes?
Yeah, absolutely. In fact, that's really my expertise, is working with space-based observatories. So the Event Horizon Telescope is a network that's at the moment completely on the ground. But my role in that collaboration is actually a part of what we call the Multi-Wavelength Science Working Group, which means that during the time when the Event Horizon Telescope is looking toward these supermassive black holes, we also harness any other resources we can get our hands on to look at the black hole at the same time. So Hubble cannot actually see these black holes very well. It can take pictures of the galaxy out at larger scales, but it turns out that all that gas and dust that's between us and the black holes actually blocks out optical and ultraviolet light. So the kinds of wavelengths of light that Hubble can see are not actually, they're not able to access these small scales for the black holes. But for X-rays, we can monitor, and for a lot of the other sort of long wavelengths, radio and other wavelengths we can monitor, and very high energy gamma rays and extremely high energy emission we can also look for. And one of the things we're hoping to do is sort of referencing back to that previous question, if we can watch variability at X-ray wavelengths from space, at very high energies, at other radio wavelengths, maybe we can use those to help us inform how we do our data analysis for the Event Horizon Telescope data. And maybe we can use all these things in concert together to get a better picture of how the jet and the accretion flow and the black hole, how all those things, those different parts interact together near the black hole. So I am mostly a space-based observer. I use the Chandra X-ray Observatory most of the time. X-rays don't come down to the surface of the Earth, so we have to do that from space, which is really good for our biology. Otherwise, we would all be irradiated all the time. But so space-based astronomy is very cool. I use the Hubble for other things, not for this particular science case. And there's a really amazing new observatory going up, we hope in about a year's time, called the James Webb Space Telescope, which I think will also be very interesting for studies of Sagittarius A star, in particular for the stars orbiting around it, like the Nobel Prize was given for this year. So absolutely, space is awesome. We hope eventually actually to put an arm of the EHT in space so that instead of only having the size of the Earth as our baseline, as we call it, we actually might be able to link up to a satellite in space too that could give us an even longer baseline, but that turns out to be really hard. So stay tuned. That might be what your generation manages to accomplish, maybe not mine.
So if you could extend like out higher, would you be able to kind of see around the objects that are blocking? Like,
Oh, not that far.
No, okay.
That would be amazing, but no. These are just things that are in orbit. So basically, the way the Event Horizon Telescope works, you have to sort of think about, so telescopes are mirror, we often make them as mirrors, and mostly that's just to collect the light. So think about the Earth, and then imagine that you only have like 4 or 5 little patches on the Earth that can actually collect light. So it's like a really crappy mirror. So it's like if you were trying to look at yourself in a mirror and you could actually only see the reflection at like 4 little pinpricks, right? So maybe you could reconstruct the image of yourself, but it would be not as good as if you could fill in the whole thing. But we will never panel the whole Earth with telescopes because people wouldn't like that. But we can try to get better images by putting strategic telescopes in at certain places and including ones that are out in outer space, which just make the whole thing larger for us. And that allows us to get that fine detail in very close to the black hole. So it would be in orbit around Earth. So you wouldn't be able to look out around objects necessarily, but it would give you sort of like a larger telescope. It turns out that's very tricky because you have to know exactly when the photons arrive in space and when they arrive on Earth and be able to synchronize them. And you have to have very, very, very good clocks to do that. And we don't quite have the clocks we need to be able to synchronize to the precision that is required for these experiments between a space-based observatory and a ground-based observatory. So it ends up being that the engineering challenges are really just really amazingly hard and interesting. So lots of smart people still thinking about it.
Thank you.
Yeah. There's one more question from an anonymous attendee. They said, your presentation is really inspiring, especially for women in STEM. I love how you related it to social issues right now and how you strive for a more inclusive community, and I agree with them. I'd like to ask if there are opportunities for undergraduate students, such as internships for astrophysics researchers.
Yeah, so there's lots of great stuff. So I don't know exactly, every institution has its own, we call them SURA, Summer Undergraduate Research somethings at McGill. Those are our institutional ones, but also NSERC here in Canada helps to fund summer researchers. So keep an eye out for those. At my institution, we post a bunch of them in like maybe February timeframe, and students can apply from all over Canada to participate. And then of course you can always email faculty. They're also, McGill is not super involved, but what are they called? They're called, they're term long, some of you will know the right name. But they're term long sort of research experiences where students go and spend sometimes a term working as undergrads at another institution. And so those I know can be very rewarding as well. So I suggest reaching out to faculty members who you think do science that you're interested in. We often work, I think I said this at the beginning, I have 8 undergrads in my group this term. In part, that's because many of our students have research requirements to complete their degrees. And right now, so many of our labs are shuttered because of COVID. So people really need sort of data intensive things that they can do remotely, science research projects they can do remotely. But I work with lots of undergrads and I think there's lots of great opportunities out there, but you do have to be a little bit sleuthy, go out and kind of look around and see who's got opportunities. But there are lots of astronomers in Canada. We have an amazing astronomy community here. So I study black holes. There's 2 or 3 other scientists, Greg Sivikoff and Craig Heinke at Alberta, and amazing Sarah Ellison and Sarah Gallagher. I mean, there are so many. Sarah Gallagher is like our current science advisor to the Canadian Space Agency. We have a really, really robust astrophysics community of researchers here in Canada. And so if you're interested in staying in Canada, definitely look around. I should say, if you're willing to go to the States too, the National Science Foundation in the US runs a series of summer programs, which I am also spacing out the name on, but look for those. They're competitive, but they often are really, really cool. Some of them send you to Chile, some of them send you to different institutions around the US. Lots of great research opportunities for undergrads, for sure.
So I think Jackson has one more question, and then we'll move to the panel.
Okay.
Going back to the start of your presentation, when you talked about your transition from studying philosophy and then going and working in Silicon Valley for a bit and then moving on to your master's and PhD, how has your foundation of study in philosophy impacted your approach to research now in astronomy?
Oh, that's a good question. So I think the program that I was a part of as an undergrad is very, very unique. So it's a small institution. It's called St. John's College, and all of the students, there's only one major. There's no different majors. It's just one single curriculum that everybody participates in. And we did a lot of reading and a lot of writing. And it turns out, I think those are the most, and a lot of presenting, a lot of talking. We had these things called tutorials. All of our classes were very small, like 12 students, and we had to read and prepare and present all of the material all 4 years. So our professors were there as kind of guides, but mostly we had to always read, come prepared, and work through the material together. And so you do a ton of reading, a lot of presenting, and a lot of writing. And it turns out those skills are absolutely crucial for scientists. So scientists write like mad fiends. We write proposals, we write presentations, we write papers to share our science with the community. We present like this, we teach. And we have to make good arguments and present them in a way that in many cases the public can understand. So you might notice that my talk has a very sort of, it's really intended to be accessible to everyone. And that's something that I think astronomers are particularly good at. We do a lot of outreach. So my degree, I think, prepared me very well to do quite a lot of compelling writing, you know, sort of trying to make good arguments for people, and also just to talk and to present my ideas. And I think those have been really, really valuable to a science career, and they're training that many of our scientists have to kind of collect on the side. We don't do a great job, I think, as science faculty and as programs to train those sort of more professional skills, the reading and writing and presenting stuff. I think that's changing, honestly, but I really encourage you, again, this is sort of harking back to your other question, I encourage you to try to expand your sphere a little and do outreach and do scientific writing. Try writing for your paper, your institutional student paper or whatever. Those skills just turn out to be very, very valuable to you as a scientist as you go on.
Thank you. That was an amazing presentation, and I'm sure we all learned a lot from you. It's time now to move on to the panel, so I'm going to hand it over to Alexandre, and he will introduce our panelists.
Hi, Lauren. Hi, everyone. Thank you, Lauren, for running the speaker session. That was really great. And thank you again, Dr. Haggard, for the great presentation. That was really appreciated. All right, so I'm going to be running the panel session. So the idea of the panel is we have 5 panelists, and I'm really excited for all of them to be here and joining us today. So the idea is just for us as the attendees to ask questions and see and learn from what these panelists have done in their research, and how students can become involved, and just kind of like learn from them. And it's more of a period where we can, more casual, where we can just have a good conversation with some researchers. So just some housekeeping items. It's a little more difficult to do this over Zoom, just because like we have 5 panelists, so one person might talk over another. So we thought it would be best if we kind of make an order for panelists to answer questions. So I just put it in the chat right now. And if you don't want to answer a question to any of the panelists, just say like pass, or the next panelist can answer, just to keep stuff from people talking over each other. So I guess if you have a question, just like what we've been doing for the speakers, raise your hand. But I'll kick us off with the first question here for the panelists. And I guess that's, can each of you introduce yourself a little bit and describe your academic path? For example, like where you studied, what university you went to for an undergrad, and what you're doing right now.
Hi, I'm afraid I can't tell if my video is working. Can you tell that, Dustin?
No, it's not.
No, it's not. I'm not sure why. I do have it turned on. Do you have any ideas here? I went in and did ask to connect it to video. I don't know what's going on here. Video settings, maybe? I apologize.
Oh, you might have, oh, no worries. You might have to go down the bottom left and click on video and say join with video.
Okay, I had done that, but I'll try it again. I had assumed that once I took off that sort of mute button and everything else, I'd be okay. I'll try here.
Janice, I might have a suggestion. When you have the start video selected, there's also a little up arrow to the right that says select a camera. Maybe that would help. Maybe you don't have a camera selected.
Okay. No, it seems to be selected. Dennis, I don't see you either. Are you on video? No?
I am now.
There you are. Hi, Dennis. Hi.
Hi.
My apologies. I'm going to go ahead and answer and play with this while we move to other people. Sorry, Dustin, could you repeat your question? Was it just about where we've started and how we got our start off in our careers?
Yeah, and sorry, I'm Alex Dodd. Dustin actually was unable to make it here
I'm
today.
sorry.
He just had something come up, so I'm filling in for him.
Okay.
Yeah, but
Great.
yeah, that's all it was. Yeah, I just want to let you know.
Yeah. All right, thanks. Okay, so yeah, my start to my career, I actually, on a complete whim, moved across the country to start a marine biology degree at Dalhousie. Because I had started scuba diving when I was 16, and I got completely enthralled with the whole concept that there were things living in an environment that I couldn't live in without all kinds of things to support me, air breathers and buoyancy compensators. And I was really curious to learn about the organisms that live there and figure out how we might be able to understand them better. Initially, I thought it would be really cool to work at an aquarium and bring organisms in, understand what they needed to survive. And so I started my undergrad degree, and it kind of one by one doors opened. I had no intention of ever becoming an academic. I didn't even know what that meant, to be honest. And I had spent my entire high school time so focused on sports, I had never really applied myself to school. And so it all just sort of fell in place. I found this interest in the marine environment, decided I would try that instead of following sports and kinesiology. And once I got out to Dalhousie, they had lots of hands-on opportunities for learning, getting in. I got into a research lab, which was what really changed things for me. And one thing after another, I started an honors project. First, I got a job at Fisheries and Oceans. Then I started an honors project that led to a summer position at the National Research Council, an offer for a master's, which rolled to a PhD. And the next thing I knew, I had a PhD. And so what I ended up doing my PhD in was biological oceanography. That's sort of taken me many places around the world. From there, I went to UBC, postdoced there, studying marine viruses, and realized that I was really getting back into a lot of biology again, and came to UNB in 2003 to start my position teaching biology, bringing in a bit of a marine twist, but also continuing to spread out as I get intrigued by new things. I would call myself a jack of all trades, master of none. And if something grasps my interest, I follow it. So that's where my career has started and brought me to today. Is Jose Moran Mirabel on?
Yep. Yeah, I'm here.
Okay, great.
Yeah, so I'll go ahead next. So first of all, I wanted to thank you guys for the invitation to participate in this panel. It's a pleasure to be here. About myself, I'm originally from Mexico, so I started my career there. I started as an engineering physics student, and I come from a family of academicians, so academia was not necessarily foreign to me, so I had that privilege of being already immersed in academia. But I'm the son of a theoretical physicist, and I did not want to follow theoretical physics. So I went into the more applied sciences. So I did engineering physics as an undergrad with a diploma in biotechnology, because I was always interested in the interface between physics, chemistry, and biology. And so after that, I did my master's at the same institution at the Tec de Monterrey in Mexico, in biotechnology, to expand a little bit more of knowledge in biological sciences. And then I started looking at opportunities outside of Mexico. And so I had the opportunity to visit a few universities before actually applying to graduate programs in the US. And I was fortunate to be accepted at Cornell University to do an applied physics PhD. And that's where I work with Professor Harold Craighead, who had a very interesting program on nanobiotechnology. And so I was always interested in the smallest scales, and that continues till today, and how they can be used to interface with biology. And so I did my PhD there, and then I stayed at Cornell for a postdoc, but I switched from the applied physics department to biological and environmental engineering, where I started applying what I had learned about microscopy, about nanofabrication and nanotechnology, to looking at enzymes and how they break down cellulose, and just trying to understand the fundamentals there. And so that was part of my postdoctoral work. And then after that, when I applied for positions, for academic positions, I was fortunate to be recruited to McMaster University. And so here at McMaster, I continue my work on the very small scales at the micro and nano size. And my group now in the chemistry department works on developing new micro nanostructured materials and applying those materials to different problems, whether they can be sensing problems, whether they are more fundamental, trying to understand how cells interact with materials at a very small scale. And so my group is a group that's composed of 6 graduate students, about 5 to 8 undergraduates, and a couple of postdocs.
Hello, Alexandre. Thank you very much for asking me to come along to join this panel. It's been an interesting conversation so far with the panelists that have, or the speakers that have preceded us. And I think we're going to have an exciting time for this next hour with the people involved too. So a bit like Dr. Miran Maribel, my start was actually in engineering physics. And maybe I'll start by saying how I got there. When I was in high school, I went to my high school guidance counselor for an appointment and asked what I should be doing for deciding on a career path. And I said that I had a real interest in doing physics. And he sort of looked at me and said, "There's no jobs in physics. You have to be an engineer." So I said, "Fine, what's an engineer?" He goes, "Well, it's like a physicist who does practical things." So I said, "Okay, I will try to follow that path." And I went into the engineering program at University of Saskatchewan. And I did about 2 years of it, realizing that I'm not enjoying myself at all as taking the conventional engineering stuff. It's not where I want to go. And I was thinking seriously, I had to drop out of it and just join arts and science physics to really meet what I wanted to do. However, just like I said before, there was an engineering physics option at Saskatchewan. So I could actually make a nice smooth transition into the physics stream through that program. For quite a while, I kind of dismissed the engineering side of what I had studied and said that was never much use to me and never very much of interest to me. Lately, I've been reevaluating that, and I'll get to that in a moment, which speaks a bit to what some of the other people who've presented have already said, that mixing your disciplines, not just necessarily within science, but between science and engineering too, can be very profitable and can lead to things that you would never get to by studying one or the other of those paths alone. So from University of Saskatchewan, doing my undergraduate, I had a wonderful opportunity after my 3rd year to go to a big nuclear physics, particle physics facility in Vancouver called Triumph. They offered these wonderful undergraduate sponsorships or fellowships that you could take there and work with a group doing interesting research on particle physics. I did my summer work there, and I was really grateful for Dr. Youngmoo Shin, who had hired me to do that. And then started to think, well, you know, doing research is actually a pretty interesting thing to do, and I think I'd enjoy it. So from there, after my undergraduate degree, I had sort of a career bifurcation I could take. Nortel Networks, which was a big optics telecommunications company, they were offering me a very nice job in Saskatoon to work with optical fiber and particularly dehydrate it to get all the water out of it, because if you have water in your fiber, it absorbs infrared light, and that's the frequencies of light that you transmit for telecom communications. Or I could go to graduate school. I was accepted at a few places and was kind of keen on going to University of Guelph to study really fundamental atomic and molecular physics. And it was a very hard decision to make as to which path to go on, but I thought that if I don't make the jump into the academic stream and try out research, I'll never have an opportunity to go back, at least not easily. So that's the road I take. I went to Guelph. I had two wonderful supervisors, Dr. Bob Brooks and Dr. Jim Hunt, who really showed me a lot about how to think about science, how to be a practicing research scientist, and they showed me the excitement and enthusiasm you can get by doing something that seems impossible when you first start it, and piece by piece, breaking apart the little bits that get in your way, and then suddenly in front of you, you've got a solution and something really interesting to talk about. And a bit like was said a few moments ago by Dr. Holland, communicating these things is vitally important. Not only communicating them well, but communicating them with enthusiasm. That seems to be a real key to making sure that you can keep your life and career regenerated all the time. The path then goes from graduate school at Guelph to a postdoctoral appointment, research associateship at the National Research Council of Canada, and then to the University of Toronto, where I did two very different things in some ways, but related in another. You see, what had sparked my interest in undergrad was quantum mechanics and optics. So a bit like Dr. de Beermacher in the first instance, and a bit of optics, a bit like Dr. Holland in the other. So at the NRC, I was studying molecular physics, using lasers, using other instruments to probe how molecules worked inside. Whereas at the University of Toronto, I did what we call quantum optics, not so much focusing on the properties of the atoms and molecules, but what they do to light and how you can exploit and manipulate that. When I got to my first faculty appointment at Mount Allison University, just down the road from us from Fredericton here, I had to make a decision of which way I would go, and I chose the molecular physics route, thinking that it would be more something that the undergraduates at Mount A could really get into more easily. I was probably wrong, but nonetheless, it still worked out to be a good path to move along on. And I continued that by being appointed then a few years later at UNB, where there's a good, strong molecular spectroscopy community. And that's where I've been ever since, studying things like what molecules exist in the interstellar medium, what molecules exist in the atmosphere of stars, using spectra I take in the lab to help astronomers identify what's out there in the space, and also to see what those molecular spectra can tell you about the conditions that they're in outer space. For example, we're doing a project with collaborators in Lyon, France, in iron hydride, where what seems to be one color of light absorption in the laboratory looks like this and spreads into several when you look at it in the sun. And the spreading is due to the fact that just above a sunspot where they observe this iron hydride molecule, you have very strong magnetic fields. And these magnetic fields interact with the molecule and split apart its spectral lines. By measuring carefully how far these splits go in the lab, we can then turn that over to the astronomers and say, the magnetic field over this sunspot on that day was so strong, and go with that. Now, I've had a bunch of graduate students and a bunch of undergraduate students who've really helped me out a lot in this research and have contributed immensely to it. It's funny, but the way it's worked out for me, I've had a lot of the undergraduates, maybe it's my early Mount A orientation, who've done enormous things in the lab and have really been helpful. Two of them, just last year or the year before, helped me to revive an instrument that was 12 years old sitting in the lab. Actually, it had been built in 1980, so it's older than that, called a Fourier transform spectrometer. So after its life at UBC, it was shipped to us, sat for 10 years in the lab. These two students decided they wanted to revive it, and by gosh, they did. This thing is now a mainstay of research in the lab, and it was an undergrad initiative that really made it happen. So I'm really pleased that they've made such important contributions to my research so far. I'm looking forward to see what the future brings with them, with me, with everybody who's in contact with the molecular physics lab. And on to Dr. Galabois.
Hi, my name is Molly. So I'm a graduate student here at the University of New Brunswick, and I did my undergraduate degree at UNB as well in kinesiology. So I guess just a little background of my research history. So I started getting involved more in the research in I'd say like my last year of my undergraduate degree. I started working in the cardiometabolic and exercise laboratory under Dr. Daniel Bouchard, and really just fell in love with research there, just kind of being able to apply all the skills that I learned throughout my undergraduate degree, and doing them in real life felt really cool and special. So wasn't really sure what I was going to do post grad. I was thinking, like most kin students, maybe physio, maybe OT, and then got involved with some projects through the lab and just didn't really want to leave them. So I decided to do my master's degree here, working on a different couple of projects. So my main thesis is physical activity and aging. So looking at different interventions that can be implemented in long-term care facilities to reduce sitting time or sedentary behavior, and measuring like clinical outcomes associated with those. So that's been like the main bulk of my research. I've also been involved in other projects looking at breast cancer survivors, and again, interventions looking at the variability with fatigue and recovery. I've also been involved in some community-based fall prevention exercise programs, so just working as a research assistant on those. And then this year, I am one of the recipients of the Canadian Frailty Network Fellowship. So this is an interdisciplinary fellowship that allows the recipients just to work with different organizations. So they funded me for my second year of my master's, which is what I'm in right now. And as part of that, I've been able to work with different organizations, including NBIRDT on campus here. So that's the New Brunswick Institution of Research Data Training, as well as some other companies, so with GNB as well. So kind of getting my feet in the door in different places. And then, yeah, so that's kind of where I'm at and what I'm doing right now. I'll just leave it there, and then I'm sure there'll be questions later.
So, hi everyone. I hope my sound is clear. I have headphones on, microphone. Hopefully that facilitates things a little bit better. So thank you for having me. My name is New. I think I might be a little bit of a odd person on the panel, a little bit different, because I actually am currently working full-time at the university as a staff member. I am a microscopist at the microanalyst and microscopy facility that's in Bailey Hall. So I'm responsible for all the biological samples that come in, preparing the samples, taking the pictures, sending the pictures off. So I work, and I also do my graduate studies part-time. So I straddle full-time job, student, and doing all the things. So the other thing that is maybe a little bit different in terms of my path versus everyone else's path is I didn't really have much of what I would call a traditional path. Or I started being interested in research as early as high school, because I would be in my biology class and I would learn different things about anatomy, about DNA and all of that. And there would be little tidbits of things that questions would pop up in my head, and I'd write these questions down in the margins. And that's how that curiosity started, to say, oh, well, this is probably something that I would end up being interested in. And then I had the opportunity to do, I don't know if any of you guys know this, but a high school opportunity. It's called Sanofi Aventis Biotalent competition back in the day. I think it's called the BioGenius competition, and it's the opportunity to come up with your own research question, research proposal, and to partner up with a professor to work on that for 4 months, and then present that, and then compete in higher levels of competition. So that's where that all started. But I didn't get to go and do research as an undergraduate student because I got NSIC, because NSICs were prohibitively competitive and my grades were just definitely not good enough. So what I did instead was I talked to I was in genetics at the time, and I talked to a TA saying, I really want to do research, I had reached out to another professor and didn't really get my foot in the door there. So I talked to her and I said, I'm really interested in doing research. And she's like, why don't you talk to my professor? So I talked to that professor and said, can I volunteer in the lab? And she said, yeah, sure. So I got the opportunity to volunteer in the lab, even though I didn't have funding that came in with me. So if you are a student that might not have the best of grades, that doesn't mean the door has to close for you. And that's why I wanted to bring that point up specifically. But my undergraduate was really characterized by, I did a Bachelor of Science with a specialization in genetics at the University of Alberta. But I would actually describe my degree as more, I was interested in learning all the skills I could possibly learn for research, because I knew that when it comes to research, and I think a lot of the stories that we've gotten to hear today has a lot of themes of you start in one area and you go into another area, then you use things from previous areas and you get the synthesis of new fields or new ideas and new pathways to go down on. That's the beauty of research. Research to me is very much a language of being able to come up with a good hypothesis, all of these different skills that you can use, the data analysis, and all of that brought together. So it doesn't actually really matter your discipline. There's a common language that you can speak and that you can take skills from one lab and transfer them to another lab. So in having that as my mindset for my degree, I focused on taking as many lab courses as I possibly could. I took lab courses in genetics and microbiology. I tried to get into the anatomy course, but couldn't get into the anatomy cadaver course, which would have been cool, but alas. I did lab courses in microbiology. I did lots of genetic lab courses, a lot of molecular biology lab courses. So everything that allowed me the opportunity to learn hands-on techniques. And if there was opportunities to do research courses where you actually work on a small thesis, then I also did that too. So I did that in my 3rd year, I did that in my 4th year. Coincidentally, I had talked to my biochemistry professor saying, hey, I really want to do the biochemistry lab course that's only for honor students. Can I do it anyways? And she's like, yeah, sure. But also, by the way, I have this extra spot for a research abroad opportunity that's just hanging out here. No one's applied for it. And I was like, what? Can I go? So I got money to go and do that. So I got to do some research in Germany too, on another topic, and then some stuff in the microscopy lab. So how did I get from this mismatch of like all these lab courses, and how did I sort of get to where I am now? And what had actually happened was my undergraduate, my not third year, but the research I was doing back in my 2nd year that had continued basically through that entire undergraduate degree, I started doing a lot of histology stuff because I worked in a lab that worked on mice. And from doing the histology stuff, and I'd have to go and take my slides down to the microscopy lab at the U of A and get them stained. And I just really enjoyed finding out what the microscopist there, Aline Otway, was what she was working on, because she always got to look at really cool things. So I just come and pay her a visit. Oh, hey, what are you working on today? What are you looking at today? Oh, what are we looking at on the scanning electron microscope or the transmission electron microscope? And through developing this relationship, that was how I ended up doing this lab course in some microscopy stuff, and then getting a summer position that's not funded by these government funding things, but they've managed to find some funding to split me between a professor and the microscopy facility so I could get some hands-on experience there too. Because when the opportunity came up for a summer student for the first time in years, they're like, oh, you want to do this? Obviously, you're going to be the first person we ask because we talk to you all the time. So that you never know where these things will lead to. And from there, I then did I worked with that professor again for another summer, doing some stuff on sponges, which then that position had to end due to lack of funding. So then I looked for another position, and there was a medical geneticist that was looking for someone who had experience with mouse husbandry and a bunch of microscopy work. And I said, okay, I'll be your technician. And then worked there for 8 months. And then one of my professors who knew that I had a microscopy background, who is the previous professor of my current professor I'm working underneath right now, because so my professor, Dr. Brian Crawford, worked for Dr. Dave Pilgrim as a postdoc. Brian sent over the posting for this position here at the UNB. So Dave said, you should apply to it. And I was like, I don't even have all the experience, you know, they kind of want this, and I have like this and this, and Dave's like, just apply anyways. What's the worst that can happen? So I applied anyways, didn't hear anything for months and months and months. And then I got word, did my interview, and I can go into, you know, those details later on. And then moved here because I got the position here. And also because I work with a great boss, and the university is very, they offer tuition benefit too. I take advantage of the tuition benefit to also do my graduate degree. So that's sort of how I ended up here. It was this mishmash of one door opens, go and do that for a little bit. Another door opens, go and do that for a little bit. Because it's better to take advantage of opportunities than to always wonder what could have been. And you never know where that opportunity will lead you next. And so that's the long winding pathway. I talk a lot with my hands, I'm sorry. The long winding pathway side of things. And in terms of my graduate research, so I work in Dr. Brian Crawford's lab. We work on matrix metalloproteinases, which are the proteins that degrade the extracellular matrix or the scaffolding around the cells. So when your cells decide they want to go somewhere else, like in cancer, then they have to make space for themselves so that they can go somewhere else. Or when you look at wound healing, for example, all of the surrounding tissue that supports your cell has to be healed, regenerated. So we look at those kind of processes by looking at these proteins, and that's where my research is centered on. So anyways, that was kind of rambling. I'll end it there.
Thank you, New, and thank you to all the other panelists. That was all really interesting learning about your backgrounds. So I guess just for the next round of kind of answers to questions, I'm just gonna see if anyone else has, or if anyone has any questions for the panelists. I think Jackson Weir had a question. He has his hand raised, so we'll see what he has to ask.
Thanks, Alex, and thanks for those great introductions. So COVID-19 has clearly had a significant negative impact on so many people across the world, including researchers. But I also think large stressors that change the way we do research, for example, going virtual, can force us to take a step back and rethink some of our scientific questions and perhaps take our projects in new directions. So my question is, how has COVID-19 impacted your research? And is there anything positive you can extract from the drastic changes that we've seen in the last several months?
I suppose since I'm the first in the list again, then I and I apologize. It turns out you're not allowed to run Teams at the same time as Zoom. Otherwise, it won't pick up your video feed. So here I am. The changes that it's brought about, I could have been much more negatively affected, except that my research happened to be in a place that was fairly easy to move online rather than in the lab. So in two aspects. So I kind of have split my career as of my research areas. Once I got it to UNB, I studied viruses in the marine environment and discovered after a few years of being here that it's extremely difficult to do in isolation, to be the one and only professor studying that area and having no colleagues to collaborate with. The amount of infrastructure that's required to do virus experiments and maintain the hosts and everything else is really challenging. So I had already moved that research over to Norway. And Norway, fortunately, has done an amazing job of managing the COVID situation. So their labs are somewhat up and running. And I have a PhD student there who's looking at virus and host coevolution, for example. And so it's interesting that there's actually, as you can imagine, increased interest in that area, given the pandemic that we're facing. So that's been sort of a benefit part to the whole COVID situation that we never would have imagined. We work on viruses infecting single-celled organisms. So we can look at models of evolution that we have a huge amount of control over, and we have organisms that we can grow rapidly with huge replicates. We can actually look at evolution happening in real time between the virus and the host, because they have such short lifespans and growth rates, rapid growth rates. So that's sort of been really interesting. And so that work is, I haven't been able to go back to Norway. I sit on these types of Zoom and Teams calls with the group back there. But the work is continuing, and I don't think we're going to have too many challenges getting funding given the era. My other angle, what I started doing when I realized I really needed to move my virus work elsewhere, I saw a growing problem in New Brunswick with an increased number of cyanobacteria blooms, or at least concerns, in local waterways, and had just started a very simple project to look and see if I could detect the genes associated with toxicity. And thought, hey, that's going to be nice and simple and easy. And lo and behold, we ended up with a completely unprecedented event on the Woolastook River that runs the entire length of the province. Dogs dying, a new toxin discovered. This all happened within my first year of deciding I was going to dip my feet into the cyanobacteria world. So talking about, paths of, taking up opportunities when they come, I ended up on this new path of studying cyanobacteria populations and trying to understand what controls them. And we've done an immense amount of sampling in the last 2 years to try to figure out where the toxins are, how abundant they are, where they're coming from, how do they move, and we have our freezers full. So while we were still able to do some sampling this summer, we also have an enormous amount of material that we are in the process of getting massive sequencing done on. Some of it's already completed. And that work then puts us into a place where we sit at computers and mine through genetic data, looking for example, we're pulling out the biosynthetic pathways that the toxins are being created with. We're looking for other novel pathways that might be there. We're looking to see, it seems like these organisms are actually able to use inorganic forms of nutrients. So we're looking for the nutrient conversion pathways that may exist. So we're actually really set up at this point to sit back, wait till we can get in and do some intensive hands-on work, and instead mine the reams of genetic data that's available. We're really fortunate that biology is in a position now, as a massive field, that there is a lot that can be done computationally. So I still have an honors student. I still have, I have a postdoctoral fellow, I have a PhD student, and that can all continue because of the amount of work we can do in silicon. Whereas I would say, if this was 10, 15 years ago, we really would have faced a much more challenging situation right now. Yeah.
Yeah, I mean, I think my research was definitely impacted, especially at the beginning of the pandemic, because we do a lot of synthetic chemistry in the lab. We do a lot of micro nanofabrication in different facilities and imaging with electron microscopes and optical microscopes. So at the beginning, it definitely impacted the students. Because we had to shut down the university, we closed our lab very quickly. And then students had to sit at home and work on their computers, and try to move some of their projects along. I think that opened an opportunity to not just sort of look back at the research that they were doing, but also look back at how we were doing things as a group. I think that this, situation where we've had to be isolated has given us the opportunity to connect more as people. And so we've started doing a lot more in terms of group activities online, remotely, but trying to keep everyone in a positive frame of mind and also, you know, hopeful for what was coming and what the next steps were. And so from that perspective, I think a positive part is looking back and really connecting more as people within our research group. On the other hand, the shutdown and the new opportunities that came with this pandemic also made me look at different areas where we could apply the things that we were already doing. So for example, we are a group that designs small molecules that we can put on cellulose, and we started this work because we were interested at looking at them with a fluorescence microscope, so we can label them with fluorophores. But we've now refocused some of that expertise into trying to graft things that might make fabric antiviral, for example, or antimicrobial. And so there have been some research opportunities that have arisen from this pandemic. On the student side, one thing that I've seen is that they've become so much more efficient. They are much more careful at planning what they're going to be doing in the lab, because they don't want to be in the lab that long, or maybe they can't because we're working in shifts. And they really pick and choose what they're going to do. And I think that that's a great skill to have, to identify the key things that you need to do to complete your project or to move your project forward. And so these are just some of the things that that have happened. Now, I think that the biggest challenge for us as a group has been with incoming undergraduate students. So we had already 3 undergraduates that were slated to start their project either at the beginning of the year or halfway through the year, and they couldn't access the lab. They couldn't be trained at the beginning. And so that posed some challenges. But the way that we've been working around it in my team is that we've paired them with graduate students that already know what they're doing. And so they're looking at a research question and maybe doing some literature research, and then participating in all the one-on-one interactions with those researchers, so that even though they're not doing the work hands-on, they're still immersed in that discourse, and they're still immersed in that exchange of ideas and troubleshooting that happens whenever we talk about research. Fortunately, now our undergraduates have been allowed to go back in and be trained, and so now they're much better prepared when they arrive in the lab. They know the background of the project, they know the goals of the project, and they can just jump right in and start doing research. So there are definitely some positive things that these challenges have brought.
Yes, I was in the sort of fortunate position at the time that COVID hit of just being in a little lull between a set of graduate students that had just graduated and a set of possible summer students that would start up. So the impact that the virus had on me was actually fairly minimal. Other than maybe the one thing that sort of made me an elephant in the room of everybody who's being affected by this, most of my energy was in fact having to go shifting over to teaching and learning how to teach under the remote environment. And that's occupied a great deal of time that I might have spent in research over the summer. So that's been an impact for sure. Now, it did create opportunities for some people that were necessary to be created. For example, we were forced to cancel our experimental physics course this term because there was no safe way to run it under the conditions that we'd have to for the COVID situation. 2 people need this course to graduate before April. So we've decided to give them an opportunity to take a course that we've never really used on our books called Research Project. It's a 1-term research project course, which normally we would say you're doing your honors and you do it in 2 terms and it's a different course number. So one undergraduate student's getting a research opportunity in the lab due to that. And it's been a lot of fun. He's been investigating the background to the project lately. What it's going to do is what we call lock a laser. Take a laser and fix it to a frequency, and if it starts to drift off, put it right back and hold it there. I need that for the instrument that my undergrads a couple years ago revived, so that I can have this nice locked laser as a reference for that instrument. So that's an opportunity that he wouldn't have had and that I would have had a tough time to justify putting time into to get going with all the other things around me. So it's worked out well in that regard. Again, you know, doors closed, doors open, as a couple people have said already, and you have to just try to make the best of what's available for you to walk through it in any given moment. I think that's about all I'm going to say about it, so I can pass it off now.
Yeah, so yeah, when COVID-19 happened, obviously our labs and stuff closed down. So we definitely felt the impact of that. Personally, my intervention, my 5-month-long trial had already been finished. So all my data collection was completed by the time COVID had started. So I was lucky in that sense. I guess as a master's student, I had a lot of time over the summer when things kind of were slower to do a lot of writing alone in my apartment. So I guess I could definitely feel the effects of isolation and quarantining on a personal level. So that wasn't as fun. Luckily enough, though, here in Fredericton at UMB, our lab is back open and running. So as of, I think, the beginning of August, we've been able to come in. We have to gear up. We wear our masks, we wear our lab coats, our visors, and we do see participants coming into the lab. And recruitment for different projects have actually, we've seen like people do want to get out of their house. People do want to come into the lab, which is interesting to see. So yeah, I guess for me, it's definitely, it looks a lot different. The research looks a lot different. It came in waves. Parts of it were slow and hard. And then, I don't know, I guess just finding alternative methods to deliver our interventions as well. We're looking at, we do some things virtual, so it looks a little bit different, but we've been able to adapt and talk to the participants a lot as well, make it focus on like the client-centered approach as well. Is important to see what they're comfortable with, what's something that they would be able to participate in as well. So that's it.
Okay, so I'll make comments on the work side of things and on the research side of things. So on the work side of things, my job in the first place has always been a very, what would be the right word for this? On and off kind of position. And the reason for that is because my job is dependent on other researchers, whether they be from industry, academia, or government, having money to say, yes, I want to look at this, because some of the microscopy procedures are quite expensive. So using the SEM, using the TEM can be very expensive. And in particular, biological samples are expensive because there's also the cost of my time in preparing the samples if you as a researcher don't want to do that yourself. And a lot of times we'll have samples being sent in from other places, which is usually more than an hour, 2 hours drive away. So they have to send the samples in. So there's all of these sort of costs to factor in. So what will happen is I'll have a project. It'll take me two to 4 months to do that project, and they'll be quiet for a couple of weeks. And then all of a sudden I'll have 3 people who want my time all at the same time, and then it'll be quiet. And it has sort of this, every day is a new day. The other part of my work also includes working with students. So I have graduate students or undergraduate students sometimes who want to be able to use a microscope themselves, or who want to be able to prepare samples themselves, especially if they're doing anything histology related. And my job is to train them. So when the university shut down, so we shut down all the way to only essentials. So the only person who was coming in was my colleague to come in to maintain the machines. Everyone else had to be at home. And there is nothing in my work specifically that is meant to be done at home, because everything requires my microtome or the microscopes, all my samples and reagents. So I had to pause that completely. And I can answer questions from students if they're starting to think about what protocols they might use, but they're also paused in a similar way. So it was a very quiet time for work on that end of things. On the research end of things, luckily I was in a transitionary period where I decided the direction I was going with my master's was not the best direction for someone who's doing a master's part-time, because it was too complicated of a project, because the experiments takes 2 weeks to run undisrupted. So I was shifting to a more molecular biology direction, so that pause time gave me more time to take a break, to reset myself, and to start thinking, okay, how do I want to approach this new direction that I'm going in? So that's where that affected my 2 areas of my life. In terms of the opportunities, we have 2 honor students so that have come into our lab, and now that the lab's open, they can come in and they can work. And they're one of the honor students project is an immunology project based on looking at the spike protein of coronavirus and whether the mom can give protection to her offspring. So they're working on that as a project. So that's an opportunity that has come up. And outside of that, we take a lot of care to plan our experiments in such a way to really open up the schedule for everyone to come in and work, because we have 5 people in the lab. And also on the microscopy, like in the facility side of things, we are not allowing people to come in to sit and watch or sit and work. So there has to be a lot of distancing. So a lot of things have to be planned out more carefully than they used to, which is a skill in of itself. So that's kind of the summary on how has that impacted things.
Thank you for all the answers again. It was really neat hearing about how COVID has impacted your experience and how you've all kind of worked around that in different ways. That's really neat. I guess, so our event's actually supposed to end at 2, but we're gonna run one more round of questions here just to kind of get a little bit more involvement. So there was a question in the Q&A and that said, there's been a lot of discussion regarding interdepartmental departments. Given the recent social issues, how do you think your field can contribute and integrate in solving those issues? So, for example, relationship of science and social sciences. And you can see that question in the Q&A session there too. And if we could...
Right there, yeah.
There you
Sorry,
go.
I'll take a look. So the question, I'm just pulling up the Q&A. I can't actually see it for some reason. I don't know why I don't have anything working here. So the question is basically linking social science to science. Is that correct? Oh, there we go. I can see it now.
I think the question had like just interdepartmental research and how you can kind of work between departments, for example, like social science and sciences and how they work together in research.
One of the, I get asked to do a lot of work, public outreach, public education, given the nature of the toxic cyanobacteria I work on, for example, and even with the viral evolution work I do, I'm a little hesitant to go any further into interdisciplinary work simply because for every new linkage that's created between my science and either a government organization or an NGO, where I work with a number of NGOs, it splits my time and it seems to fractionate it even more. I think it's incredibly important that scientists communicate their findings to other groups and make them accessible and create linkages. But at the current moment, I personally am feeling like I have to, it necessitates that I do less science. And I'm not sure that's the best approach. My training is very much in science. And so I guess to give you a better example of what I mean, I recently was awarded large funds from Environment Canada, ECCC, Environment and Climate Change Canada. And they kept asking, you know, how much funding do you need to do public outreach, to develop educational stuff and do everything else? And I said, well, in my proposal, this is bare bones research. This is money simply for a postdoc to grind through metagenomic data. If you want me to do that, I have to rewrite the proposal, find new collaborators, find someone else to hire to do all of this work. I'm not trained to do this other end of things. I think there's a huge use of it being done, but I guess that's just sort of an angle that concerns me a little. I think we need to train science communicators. We need to train people who work in the interdisciplines, but it's sort of stretching limited resources really thin. Sorry, it's not the most positive answer, but something has to give at some point if you're going to take a current research program that is very heavily science-based and expect it to then become a linkaging project. And that's something to be aware of. You can't be everything to everyone. You do have to figure out where you fit, where you make the most impact. And luckily, ECCC, when I expressed my concern, I said, sure, I can do that if what you need to fund involves a lot of sort of social issues around water use and that we'll do it, but I can't then do the genetic work. They said, oh, well, no, we don't want you to stop doing the work you're doing. We just wanted you to add all this extra onto it. And I was like, well, then I'm going to need a lot more money. And they decided, no, they didn't have more money to give me. So it's sort of an example of there's a limit. Yeah.
Yeah, I was reading the question on the question and answer chat, and one of the things that the question asks is about recent social issues and how our fields can contribute to these social issues. I think being in science and technology, we can definitely do a little bit more or a lot more to increase diversity and inclusivity within our fields. I think that, as Dr. Lawrence and Dr. Haggard mentioned before, starts with good communication. And so one of the key things that I think we can do is train our students to be more proficient in science communication, being able to distill what they're working on, which might be very technical, down to the thing that is important to people, and to try to connect with the more general audience and translate what they're doing into something that they can relate to. So I think that that's one place where we can contribute. I think that on a chemistry side, I think that our research can also contribute to coming up with more sustainable ways of doing things. And so whether it means finding replacement for materials that might pollute the environment or might use toxic chemicals, for example, or finding new ways of making those materials, I think that that's another way that we can contribute and improve society. And finally, on a very personal basis and on a research group, I think that we can always strive to promote all our undergraduate and graduate students as much as possible, be as equitative as we can, and also implement some policies that allow us to recruit from a wider base of backgrounds. And so I think that these are some of the few things that I can think of that we can do to, improve and contribute to society.
Yeah, there's been some really good points raised by Dr. Lawrence and Dr. Morin Mirabel about these things. I certainly agree with Dr. Lawrence about the time fractionation problem, because I find that if the one more thing on the plate can often just tip the whole balance over. And you really can't do everything for everybody. There are people who make a career out of doing interdisciplinary work, and most of that career seems to be, and it's an important role to fill, as a broker between this discipline and that discipline, and knowing enough about them both to know what's important to take from them both, but not expecting either of the two sides to be independently at the same level as they are. So things like that restrict and both give you opportunities to do the interdisciplinary work that you're talking about. My experiences with interdisciplinary work have not been too extensive. I was involved a bit in a hydrogen fuel project based in Fredericton until the company had to close up shop. I've been asked about whether or not infrared laser light could be used to examine whether a potato has a void in the middle or not, which is societal. It affects our food supply. And I've been reading since the COVID situation about how people can use laser light through a technique called Raman spectroscopy to identify laser or viruses in environments with unique signatures that are optical. So these kind of things have got my mind going as a footstep into that world. But I did appreciate Dr. Miran Mirabel's comment about the inclusivity business, because I've been approached recently by a potential graduate student who's thinking of starting a degree in about another year's time. And they're interested in pollution monitoring using lasers and looking at the way that a laser can probe the environment in a sort of a non-destructive way and in an instant way, so you don't have to wait for lab results to come back. The prospective student has an Indigenous background, and you can say, well, you know, that's interesting. It wouldn't be something I would naturally gravitate to, but I do want to give a student the voice and the opportunity with what I have in the lab in order to pursue something that's of interest to them and of broader use to society too. So that kind of thing is a way that one suddenly sees an opportunity pop up to take the research in a different direction and move it along in the societal paths too.
Yeah, so like I mentioned previously, this year I am funded through the Canadian Frailty Network, and it's through an interdisciplinary fellowship. So the whole goal is to tie in different disciplines at addressing issues of older adults living with frailty. So we do, like, whenever we're developing different projects, they really encourage us to, coming from someone with a kinesiology background, to tie in the nursing faculty, someone who works in gerontology, just bringing all those fields together to see all the different angles that can be approached. I think that's especially important in clinical research when you're looking at different interventions to see, okay, like, well, and also engage the client. So when doing so, just to make sure that you're not really missing anything. That, for example, it might seem feasible, but if it's not something that the person likes, where will you go from there to actually implement it in the long run? And I think this was touched upon earlier as well for recruitment, just trying to get draw from people from all different kind of backgrounds so that you can generalize your findings as well. That's really important if you're just, depending again on the type of research, but if you're just kind of focusing on just recruiting from one subpopulation, it's not, depending on what you're trying to find, to generalize those findings in the end won't be as valuable or reliable, I guess.
Okay, so I'm going to answer this, but maybe take a little bit of a different direction or make a different point than the directions that have already been sort of presented. And so on the topic of inclusivity, I think that people sometimes, there's a certain separation in terms of ideals and how things are actually optimally run with the constraints in terms of money, recruiting process, or all the other things that exist. And one of the things that I've been asked before by people of color in terms of, okay, where's better representation in science? I said, I'm not saying that there isn't maybe an attitude problem that could exist at times, but also that there is a issue of, if you are in a situation where you have less resources, or you don't have the support that you need, or so on, how can you, even if you won a scholarship for an entire summer to have the opportunity to work in a lab, how can you take advantage of that if that summer is also where you need to make enough money so that you can actually fund your studies for the entire year? So I know of people who could not, they won the NSERC and said, well, that's great, I can't take it $5,000 is not going to last me the entire year when I need to go and work a job to make at least 16, 20 grand in that summer so that I can support myself the rest of the year. So it's not just a question of, what can science do as us as academics, but it's also a greater question of the policy making that sometimes we as academics get tied up by. And I don't know the answer to that, except more advocacy work to say that research is important, proper funding for students to do research is important, to equal the playing ground so that we can have a greater diversity of voices, because they're not making a choice between doing science, something they're interested in, and making sure that they can support themselves and their families if they have families involved too. So that's kind of my commentary on that sometimes it's not as nefarious sounding as it gets made out to be. There's logistical things that go beyond, which is why if as a scientist, we do science because we like to be at the bench. Oftentimes we don't really care to talk to people. I mean, we like people, but if we really wanted to work with people a lot, we might choose a science profession that really supports that, or we might choose a completely different profession. I wanted to originally become a doctor, and I eventually didn't become a doctor because I didn't like studying human genetics because it's a lot messier than working on developmental biology. But for the scientists that do have that excitement to talk to people, advocate with people, you also, if you are not one of those people, then you should support those people to do their jobs, because it is a specific role that not a lot of scientists are interested in or have the skill capacity to, or care to develop the skill capacity to do. And the ones that do oftentimes get all sorts of criticism, whether their message is not good enough, it's not inclusive enough, or they don't even like what's being said in the first place, if it's anything about GMOs or other technologies. And so if you want to see more inclusion and if you want to see more support, science needs a lot of advocates for that in the scientific community, outside of the scientific community. And at the end of the day, that is you talking to your friends and family and being like, hey, I'm your happy fellow scientist. I'm a nice person, and this is what I do, and I really enjoy it. I'm really passionate about it, and it's not so scary. And I think that helps to open up the conversation and helps to open up better support for what needs to be supported. So that's kind of my 2 cents on that. That's not really interdisciplinary, but it's trying to talk about the whole thing as a whole.
Well, thank you to our panel and all the great answers to the questions today. We're going to have to wrap it up here just because it's already past 2 o'clock and we just have a couple more things to do before the conference is over. So we really appreciated you taking the time today to come and speak to the students, and hopefully people got to take a lot out of the panel and learn a lot about your experience in research. So Emma, we have a couple door prizes for the virtual event, and we will mail them to whoever wins it. And Emma is just going to come on here. She's a junior editor-in-chief, and she'll be talking about the door prizes.
Yeah, thank you, Alex, for the introduction. So I want to say a personal thank you to all the speakers and panelists for their time. And I also want to say a thank you to everybody who took the time out of their weekend to come to the ASRJ's first virtual event. So in light of this, we wanted to have door prizes for the event. So we had a book and 2 gift cards from the Tipsy Muse and the Chess Piece Cafe. And Alex has the book, if you have it right there to hold up.
Oh, and it's authentically signed by the executive members of the ASRJ, so it's a keepsake. And it's braiding sweetgrass.
Yeah.
Yep.
So for the book, we drew Dr. Haggard's name. And for the Tipsy Muse gift card, we drew Madeline McIntry. Sorry if I pronounced your name wrong. And for the Chess Piece Cafe gift card, we drew Nicole Acupin. Sorry if I said your name wrong again. So congratulations to all the winners. And if you guys would like to drop your emails in the chat so we can reach out to you for your address for mailing, or if you guys wanted to reach out through social media, that would be great. So thank you once again, and I'll pass it back over to Alex so he can conclude the event.
Thank you, Emma, for doing the door prizes. That was awesome. So I just want to give a thank you for me personally and on behalf of the ASRJ to all the speakers, attendees, and the panelists for coming today to the event. It was really amazing to hear about all the cutting edge research going on locally and kind of across Canada, and also learning about opportunities for students to get involved, which I think is something really important to talk about. And I also want to give a special thank you to Dustin McKee. Unfortunately, something came up and he wasn't able to make the event today, but the event wouldn't have been possible without him. He put a lot of effort into organizing this and contacting all our speakers and getting everyone here to speak and kind of just organizing everything. So just a big thank you to him. And yeah, we hope you all got to learn a lot and enjoy it. So just two kind of housekeeping items. We have a feedback form just to kind of hear what you all thought of the event and how we can go ahead and improve future virtual events. This is really new to us, having a virtual event. So we'd love to learn how we can make it better. And in light of that, we're actually having another virtual event in November called ArtSpire, and it's going to be more focused on arts research and kind of trying to engage that community. So if you or anyone you know is conducting arts research or just want to attend, feel free to let us know, and we'd love to have you. So I'm just going to put, I will send an email with a feedback form to everyone, and that concludes SCIspire for 2020. Thanks a lot to everyone.